Buenos días. Vamos a iniciar con unas palabras de nuestra directora, la doctora Palomares. Gracias por estar aquí. Entonces, escuchamos a la doctora y pasamos a la primera presentación.
Muy buenos días a todos y a todas. Bienvenidos a nuestro último simposio en celebración de nuestros 40 años. Y bueno, pues vamos a cerrar el año con broche de oro. El programa está extraordinario. Muchas felicidades al departamento. Este departamento, bueno, pues creo que es de los más diversos, pero también más interesantes del instituto. Y pues parte de las líneas que se realizan en el departamento van a quedar reflejadas en las charlas el día de hoy, que incluyen investigadores de Alemania, Francia, Canadá, España y Estados Unidos. Los invito a todos a disfrutar este último simposio y también aprovecho para recordar e informar a los que están escuchando que tenemos nuestra semana académica, la semana del 12 de diciembre, también con pláticas extraordinarias. Entonces, ya que están escuchando este simposio, pues los invito también a unirse a las otras actividades académicas del instituto, como son la semana académica. Sin más, le doy la palabra al doctor Covarrubias, quien hará la presentación de nuestro primer ponente. Gracias.
Well, it is really a great pleasure to have Dr. Hans Clevers as a speaker for this celebration. As probably you know, Dr. Clevers is a professor at Utrecht University. He's a head of pharma research and early development at Roche. Dr. Hans Clevers is a unique researcher, not only because he has published more than 700 articles with more than 100,000 sites, or because he has received many prizes, among those the Keio Medical Science Prize in 2019. But Dr. Clevers is a unique for inspiring scientific career, particularly relevant for students and young scientists. He's a very good example of how, by responding key biological questions, research can evolve to reach experimental models that not only become powerful tools for studying human development and diseases, but also tools with potential biotechnological and regenerative medicine application. Welcome, Dr. Hans, and we are ready for your presentation.
Yeah, well, thank you very much. I try to share my screen. It should work now. Yeah, I assume you see my first slide now. Yeah, so what I'll try to do in the next 45 minutes or so, give you a background as to how we developed so-called organoid technology, and I'll give you a number of examples of how we have applied this organoid technology. And what you're looking here at is the inside of the intestinal tract of the small intestine. These protrusions that you see here are called villi, and these little pits, these little holes, are the crypts of Lieberkühn that you might have heard of. And it had been known for a long time that at the base of these crypts, these multicolored cells, there were stem cells. Now, I'll try to convince you in the next few slides that actually these cells here are the real stem cells of this system. So why did we think that this was an interesting tissue to find the stem cells in? Here you look at a different view of the same intestinal tract. Again, you see one villus. You see two crypts here. There's probably about 10 crypts that sit at the base of the villus. Somewhere near the base of the crypt are the stem cells that we hope to find. And what was known when we started doing this is the end of the 90s, so more than 20 years ago, is that these stem cells probably are the most active stem cells of the human body. They produce daughter cells, so that was known, and these cells are all rapidly dividing. After about 2 days, so they're born somewhere here, they move up while they divide. After 2 days old, they exit the crypts. They become one of the about 10 or so different cell types. They move up, so now they can help take up nutrients into the blood and lymph vessels. but also keep the bacteria and all the other stuff out. And when they in the mouse are 5 days old, they reach the tips of the villi and they undergo apoptosis, which means that the entire lining of the intestinal tract self-renews, so is replaced every 5 days. So this is by far the fastest system of a mammalian body, fastest stem cell system. Now, we knew that, I won't go into detail here, but we knew that Wnt pathway was very important for these stem cells, but also for cancer of the gut. And we eventually found a marker of these tiny cells that we thought were the stem cells. And Nick Barker made an experiment in mice where he could mark one of these stem cells in blue in a mouse. Over the next few days, he saw blue daughter cells. You see that here, rapidly dividing. After 2 days, they exit the crypt, the blue cells. We saw the different cell types in this ribbon. And over the next day, so day 3, 4, 5, after he marked that stem cell in blue, we now saw blue daughter cells moving up the flanks of the villi. And about 5 days after that one cell was marked in blue, the first blue daughter cells arise at the tip of the villi. You see that here, and they undergo apoptosis. They commit suicide. Now, this experiment showed two things. First of all, the one cell that we mark at the bottom of the crypt through this LGR5 gene that we make blue is actually long-lived, because if we let these mice live for 2 years, the blue ribbon will still be there. And also in the ribbon, which is produced by one cell, we see all of the other cell types, so goblet cells, enterocytes. So by our definition, these LGR5 cells are the stem cells of the gut epithelium, so the most active stem cells of the mammalian body, a human or mouse body. When we looked at these cells, we realized that they divide every day. Every about 23 hours, they go to one cell division. And in a mouse, mouse can be about 3 years old in a lab. That cell would go through 1,000 consecutive cell divisions. And when we saw that, Toshi Sato, you see a photo here of him, gastroenterologist from Japan working in our lab, and we sat down and we thought, well, maybe we can take one of these stem cells, find a good mix of growth factors, and then if it's the right mix for these particular stem cells, maybe they start dividing. And we don't do this in 2D on plastic, like we usually grow cells, but we do it in 3D in so-called matrigel. So this is collagen, laminin, so it's like a droplet that is a gel. The stem cell sits in the middle and it can form a 3D structure inside the gel. Now, when we did this, we noted, so these are the key growth factors. Epithelial growth factor, arspondin is a Wnt activator, and Noggin is a BMP blocker for the insiders. We got structures forming, and much to surprise, this is what they look like. They turn out to be small copies of the intestinal epithelium. So there is, it's a closed space that they form. It's not a tube, but it's a closed space. But when you put them together, they actually fuse and you can make tubes. You don't see that here. Here you see one organoid, one mini-gut, another one, a few more here. The central space here is a central lumen, is lined with the cells that you normally see on the villi in the gut. And these buds are the equivalents of the crypts. And so there are many, many stem cells at the base of the crypt. There are many Paneth cells in between the stem cells. And there were many surprises. First of all of the cell types of the gut epithelium are made by these organoids. And the organoid comes from a single cell. They sit in the right place. So the Paneth cells and the stem cells sit at the base of the crypt. The other cells sit in the around the lumen here, and the rapidly dividing daughter cells sit here. That was one surprise. So they really make a complete copy of gut epithelium in the lab. But also, second, they keep on growing. They never stop. We have grown them for many years now, much longer than a mouse can live. This is mouse gut still. Telomeres stay long for the insiders, and we can even transplant, as I'll show you a little bit later. And we didn't, so people predicted, well, if they grow so well, they must now be cancer cells, they must be malignantly transformed. However, if we sequence the DNA of these cells, we never have seen oncogenic mutations. So telomeres stay long, all of the cell types of the gut are there. They don't look like they're cancer cells. They really look like we are growing normal gut epithelium from a single stem cell in a lab. And these structures are now called organoids. And I will mostly talk about gut, but we can now do this, and many labs do this from many other organs. And this is a recent paper that we published. And here you can see how complex these organs can be. This is from the human small intestine. So these are the things that stick out are the crypts. So you see these dark cells, they are Paneth cells. By CRISPR, we can knock in fluorescent colors. So we knocked in green in the mucin 2 gene, marking goblet cells. So these are goblet cells. Defensin A is red. These are Paneth cells, and in purple, chromorenin A, these are the rare hormone-secreting cells that also are quite abundant in the gut. And there's several other cell types for which you also make these knock-ins. So really, this is a reliable copy of normal human gut epithelium, yet it's not in a human being. It sits in matrigel in a 3D structure in a lab. And as you can see, they're about maybe 0.5 millimeter to 1 millimeter across. Now, how normal are these really? We grow them from one stem cell. We grow them extensively in culture. Maybe they have mutations. Maybe they have become cancer cells. Maybe they are damaged. They are exhausted. So to test that, Toshi took a single stem cell from one of our mice. So this is not human. Actually, what I'm showing you now is currently done in humans in another university in Holland, in Groningen. We took a single stem cell here. We grew it up to about 100 million cells in the form of these mini guts. We then sent them to our collaborators in Japan, Mamoru Watanabe's lab, and the organoids were RFP positive, so they're red fluorescent. That's how we can follow them in Japanese mice. So they were infused. So these are the Dutch organoids grown from one stem cell. They were infused through the anuses of several dozen mice with inflammatory bowel disease. So you can see here these ulcers. And what happens is that these organoids, they float around. When they see an ulcer like this, they will go down, they will attach, and they will open up, and like a living Band-Aid, they seal these lesions. Now, of course, this is an animation. This is the real experiment. Here you see a mouse colon. So this is a Japanese mouse. Everything is black, but you see an orange-red patch. That's where the Dutch cultured tissue has inserted itself. And I stress, so this was done from one cell, one stem cell, expanded and given to multiple dozen mice, so 40 mice or so. And you can see it's nicely integrated, and by all means, this is normal tissue. We never saw that this developed into cancer. We also never saw this getting lost because the stem cells were exhausted. So this experiment, which is a few years old, really proves that you can take a single stem cell, you can grow them, it is normal tissue, you can transplant it back and actually cure mice. And this is now for another organ, the salivary gland here for dry mouth disease is being done in humans. And for inflammatory bowel disease patients, this is currently done in Mamoru Watanabe's lab, our collaborator in Tokyo. So while we were doing all these experiments, we realized that this little trick of taking a few growth factors, matrigel, and then a stem cell from the gut, that it works for the gut very well. First we thought that gut stem cells are very special, that they can do this, but we now know that probably all internal organs have stem cells, that when you put them under similar conditions, with growth factor, the right collection of growth factors in the 3D structure, you can grow mini livers or liver organoids, pancreas, prostate. Well, you see a whole list here. Recently, we made tear glands from the eye. They can actually cry. Taste buds were made by a Japanese group. They can actually taste. They can taste well. They come from the tongue, etc., etc. So it's really surprising that you can do this. They often grow for long periods of time, for multiple years, so like cell lines. I should stress that we don't have to start from a stem cell. That would be very difficult in practice, but you can just start from a small bit of tissue from a patient, a biopsy, or maybe something that you get after surgery. We grind it up in small pieces, 10, 20 cells per fragment. We put it in the matrigel, we add the growth factors. And what you'll see then is that the epithelial cells will immediately form a space and will start growing, and they make the organoid. And all the other cells actually die. So immune cells, blood vessels, fibroblasts, they will disappear. You're only left with the epithelial cells. But if you think about all of these organs, epithelial cells really perform the organ-specific function. Say for the lung, for the liver, for the pancreas, it's the epithelial cells that perform the organ-specific function, and the other cells, immune cells, blood vessels, nerves, they support that function. But the many organs that we grow, we think really capture the real function of organs. Now, you can do this out of interest or for some basic biology question, but you can also use these organoids to model human diseases. Now that we can grow normal tissue, it's quite easy to grow cancer. So basically, you take a tumor directly from a patient, I'll show it a bit later, and grow it in culture in the lab and do experiments. You can do it from patients with hereditary diseases. I have no example today, but we did this extensively, for instance, for cystic fibrosis and some hereditary liver diseases, where you have the hereditary model in the lab that represents an individual patient by getting a little bit of tissue and grow it in the lab. We call that an avatar of the patient. And you can also give some examples. You can also study infectious disease quite well in these organoids. One example of a different organ, the liver, again, epithelial cells. So the hepatocytes are the chemical factories of our body. They are very important for metabolism, as you know, but they also make most of the serum proteins in very large amounts, albumin, alpha-1 antitrypsin. Bile ducts that you see here are formed by cholangiocytes. They are the other important liver cell type. And the rest of the cells, blood vessels, Kupffer cells, stellate cells, etc., are not real liver cells. They migrate in, but then don't come from the endoderm. So there's only two real liver cells: hepatocytes, cholangiocytes. Now these we can also grow. Cholangiocytes are quite easy. We sort them out. They're EpCAM positive. They build the bile duct. They're beautiful cells. But when you take one of these cells, you put them in a drop of hydrogel, of matrigel, and the right growth factors, they immediately make organoids. So they become bipotent stem cells when you put them in culture. Here they are differentiated cells, but in 2 days they become these cells. And now they can become both cholangiocytes and hepatocytes. And the hepatocytes, you see the word here, are the cells that make albumin, have all the enzymes that we need for metabolism and for drug metabolism. But they have always been very, very hard to grow. Now that was solved by Hu Li Hu quite recently. So here you see first in mouse, one large hepatocyte. They're really very large. They're 40 to 50 micrometers in diameter, large nucleus, often 4N or 8N chromosomes. But if you get the right cocktail of growth factors, you need about 8, published by Hu Li. And you can actually see that one cell grows out into this structure. It's not the fastest growing organoid. It takes about 3 weeks, but you can see it forms a nice structure. And if you do this for humans, again, we don't start from a stem cell. We start from one hepatocyte. You can see very large cells, very large nuclei on a single section of the confocal image here. You can see there's a very nice nucleolus, typical for a hepatocyte. We stain for alpha-1 antitrypsin, which is a serum protein, very abundant serum protein. You can see there's a lot of this secreted protein is being made. And when we carefully compare one of these cells to a real hepatocyte out of human liver, they essentially are identical when we do single cell sequencing or any other analysis. The one difference is that they actually divide in organoids. You can see here a nice mitosis, 2 sets of chromosomes here. And by any means, these are normal hepatocytes, but we can grow them long term, particularly when they come from a young donor. We can grow them for very long periods of time. And we can also, and I'll show that we can, with CRISPR again, quite easily genetically modify and make them into interesting experimental models. So here you see a 3D structure on a blow up. We also noted that these large hepatocytes not only just grow and make more hepatocytes, but they also create these structures, and they're called bile canaliculi. So ducts in which the hepatocytes secrete the bile acids. So what we stain here for is a bile acid transporter, MRB2. You can see it, actually the bile acid goes into these ducts and then comes in the central space. And we believe that normally this is where the bile duct would be, the real duct that makes by the other cell type, by the cholangiocytes, but they don't exist in these organoids. So we get these central spaces that fill up with bile acids produced by these hepatocytes. And eventually they explode and then they close again. Again, showing how complete these organoids are. They really consist of normal hepatocytes that exert the functions that they would normally exert in a real liver. And when we, so this is a knock-in, this one moves. So here actually, you see a single hepatocyte where Daizong Wang, a Chinese postdoc in the lab, knocked in GFP into the MRP2 locus, so this is the gene that encodes for the bile acid transporter, and there's no expression when there's one hepatocyte, not when there's two hepatocytes, but as soon as a few more and there's a little luminous follow, they immediately start putting the bile acid transporter there. You can see it nicely here. It grows and grows, and eventually it creates an organoid where every hepatocyte is connected to this network of bile canaliculi, and maybe this movie runs. Yeah, you can see actually how active these bile canaliculi form in a growing hepatocyte. Again, stressing that this is really surprising how well these hepatocytes, you would never call them a stem cell, they are real functional differentiated hepatocyte, but given the right growth factors, they start behaving like a stem cell and they make these structures. We can use this for disease modeling again. They can be transplanted, I'll show you a bit later. But here we modeled a disease for which there currently is no laboratory model, chronic HBV infections, hepatitis B virus infections. Because we can grow these hepatocytes over long periods of time, we can infect them with the virus and then follow the virus over long periods of time. And that could never be done before. And here we go out 32 days, but Daizong is now currently working on models where you can do this 2-3 months. And you can see that the viral presence remains stable over time. And in the medium, you can actually find that the cells consistently produce the viral particles. So this is now a model, we think, for chronic hepatitis B infections, a major problem, particularly in countries like China or Thailand and in Asia, where 20% of the people who get infected will turn it into a chronic infection, and then slowly, year after year, will lose parts of their liver function, and often this develops into liver cancer or total loss of liver function. Of course, a terrible disease, and this would be a way of possibly finding drugs that could treat chronic hepatitis B infections. That's ongoing. Here we transplant. This was done by a postdoc in my lab, Helmut Gehr, together with Ipe de Jong in New York, our collaborator. In blue, you see a mouse liver. In red, you see human hepatocytes that have been transplanted 2 months ago, and they're still dividing, as you can see. They're Ki67 positive. And they make these red islands that slowly grow into the liver of these mice and replace the mouse livers. As I said, we can also grow cancers. We can grow the normal tissue, and therefore we should also be growing cancers. And we've done this for many different types of carcinomas. What you see here is the idea would be that we get healthy tissue and diseased tissue from the same patient, in this case, a colon cancer patient. We grow on the left the healthy cells, on the right we grow the cancer cells, so they come from the same individuals, but the ones on the right will have oncogenic mutations in their genome. We can sequence, of course, directly the DNA of the cancers, but that's not very special because that could be done on the tumor as well. But more importantly, we have the live cancer cells and we have them in the lab, and we can now do interesting experiments, but we can also expose them to a series of different drugs and then see which drugs will kill or will not kill these cancer cells. And this can be done in a personalized fashion. If we get the cancer cells from a single patient and we grow them up, we can then test them and then we can find the best medicine for that particular patient. That works. Here you see healthy and cancer from the same individual. And I stress this could never be done. So there are, of course, many cancer cell lines. It's not so easy to, from a patient, develop a cancer cell line. Only works maybe 1 in 100 or 1 in 1,000 tries. But cancer organoids almost always work. But at the same time, we have normal cells, and that never existed outside the human body. So you can compare normal cells and cancer cells in whatever experiment you want. And here is from another patient, but as you can see here, we now have many pairs of normal cancer for lung, but also for breast cancer, for gastric cancer, for liver cancer, for lung cancer, etc., etc. Now, we were hoping to show that these organoids predict well what drug works for which patient. And while we were doing this, you see a few of the examples here. A paper was published from Great Britain, from the UK, beautiful paper in Science 4 years ago, that shows that indeed patient-derived organoids, so these cancer organoids, predict very well what happens if you follow patients in the clinic. And in this case, in all these 4 other papers we were involved in, since then, this is all 2019, since then there are many more papers. And essentially all of these papers show that if you run a trial and you give patients a particular medicine, and at the same time you grow organoids from that patient and you test the response of that medicine in the lab, and then when you compare the response of the organoid versus the response of the patient, it correlates very, very well in the order of 80 to maybe 90%. So if you test in the lab if your drug will work or will not work, 90% of the cases you're correct, the drug will or will not work in the patient as predicted by the organoids. Now, currently, this is still slow technology. It takes multiple weeks when you get a little bit of tissue to grow it up, do the tests. It's also very expensive. It's also difficult. You need really skilled people. But there are now a number of companies, and I am a founder of one. But so I have to mention that. But there's multiple companies developing machines now, instruments, where you would need very little tissue. You would grow it for about a week. You would then do your test. And after a week, you get your readout. And we think that given how well they predict, this approach will be quite powerful in the clinic to complement the standard diagnostics for cancer. So for cancer, basically, you do histology, look under the microscope at the tumor. Pathologist will give a diagnosis. You can do DNA sequencing, a lot of imaging, of course, but we think that you can also grow the tumor and directly test if a particular treatment, radiation also works here, so radiation or chemotherapy, see if that works, and then also help advise the oncologist to what the best treatment would be for each individual patient. This is another study that we recently published. It is Nature Biotechnology, mostly done from Anne Rios's lab. So here you see in yellow breast cancer organoids. When they die, they turn orange-red, as you can see here. And in white-blue, you see immune cells, CAR T cells, that have a particular CAR T cell receptor. And if you want to know the details, you can go to this paper. But they can recognize cancer cells and kill them. And that's what you see happening here. So slowly, everything that was yellow originally is being killed and turns red by these CAR T cells. So T lymphocytes that are given a special receptor that allows them to recognize cancer cells and then kill them. Now, what Anne's lab has done is essentially tracked individual, you see that happening here, individual immune cells, and then classified the behavior of each immune cell, each CAR T cell. Sometimes they don't move at all, like this one here, just sits there. Others run a little bit, but they all behave differently. And there's about 6 different types of behaviors. When you now zoom in on cells that have a particular type of behavior and then look into these movies, you see things like this. So here you see one of these cells attaching itself to, so it's blue-white, the immune cell. The breast cancer organoid, about 20 cancer cells, is yellow. You see that a single cell, you see a clock running here, so 14 hours, within a matter of a day, kills one by one, kills every single cancer cell in that organoid. So it can kill about 20, 25 cells in one day, all one after the other. And you see it totally destroys the cancer cell. And it's still fine after 1 day of hard work. If you look more carefully at what happens, so on the left, there's one of these active cells again. You see it immediately sticks out. It's these very thin extensions here that basically wrap around cells and very far away from the main body of the cell. And it uses these extensions. This has never been seen before. It uses these extensions to slowly destroy the breast cancer organoids. And then I think the most spectacular cell type that she's seen, Anne calls the serial killer. So it's the one here in the bottom. So it runs much faster than any other. It drills through the organoids. You can see it goes straight through. And when it's done in one organoid, it moves to another one. You see it kills another one cell here. It kills a few cells there. It then moves to a third organoid. You can see that happening here. And kills more cells, as you can see here. And now we're only 6-7 hours into the experiment. And then it disappears into the sunset. So these are called the serial killers. And Anne actually was able to isolate these because they drill, as you can see here, they drill into the organoids. So if you wait 2-3 hours and you then spin the organoids down, these cells will be inside. You can do single cell sequencing. So we know quite well what they look like. there's a few percent of the T cells behave like this. And what we think is that it would be great if one could have many more of these cells, that the patient would benefit a lot by having rather than 2 or 3% of these very active cells when they get CAR T cells. Maybe have 10 or 20 or 50% of the CAR T cells behaving like this. And it would probably be much more efficient in killing the tumors. So it's another example of using organoids where you essentially mix organoids with immune cells and now start asking interactions between diseased cells and cells of the immune system. Here we do something else. This is a paper that was also quite recently published. Here we are going to add bacteria from the microbiome from the gut onto the inside of gut organoids, of colon organoids. And the question here is the following. You have all heard of E. coli as one of the main species in our microbiome. There are particular strains of E. coli that are called PKS E. coli, and they have 60 kilobases extra DNA in their one circular chromosome. And these 60 kilobases have about 10 or so genes, and these are enzymes that together can make a small molecule that's called colibactin. And it has been known for quite some time that if one grows a PKS E. coli cell on top of your normal human cells, you will see DNA damage, double-strand break. And the hypothesis is that this particular E. coli, it secretes this substance colibactin. I'll show it in the next slide. It secretes it into the human cell, and that it will then go to the DNA and somehow damages the DNA. And so these are genotoxic. In doing so, they will kill the human cells. The question that these 3 PhD students, Axel is in Ruben's lab, Cayetano and Jens were in my lab, what they asked, maybe PKS E. coli is not only genotoxic, killing cells by damaging DNA, but maybe it can cause mutations in DNA. And that would be very, very dangerous because we know that 10 to 30% of people have this particular E. coli in their microbiome. And if indeed this would be a mutagenic or maybe even a carcinogenic bacterium, it could contribute to the formation of colon cancers. So this would then be a very dangerous bacterium. Now, this is the small molecule that is made by PKS E. coli. I won't say too much about it. It was published a few years ago. So chemically, it is known that there are 2 warheads here and here. And with these warheads, it can covalently bind to A residues in DNA helices. And if that would happen on 2 opposite strands, so it binds here, this is colibactin, it binds to an A residue here covalently, binds to an A residue here. Now there is an interstrand crosslink. This is not allowed. The cell will die unless it can get rid of this whole thing. And so that's when the double-strand breaks occur. But possibly also in trying to get rid of this, the cell actually repairs its DNA and introduces a mutation. So that was the question, are these bacteria not just genotoxic, so cause destruction of DNA, but are they actually mutagenic? Do they cause mutations in live cells? And if that would happen in your gut, would be dangerous. Now, we got a very active PKS E. coli strain from our collaborators in France. And we got a mutant PKS E. coli that lacks this particular, see that here, this particular gene. You can easily culture these bacteria on agar. We are not really experts in bacteria, but of course we know how to grow E. coli as molecular biologists. So you can pick them up, inject them into normal human colon organoids. So the insides of this organoid would be equivalent to the insides of a real gut. So here you see them inside, here the villi. And now on the left, you'll see production of colibactin overnight, and on the right is our negative control. So first we wanted to see if we could recapitulate what's in the literature, which is that if you expose these organoids to PKS E. coli, you will get DNA damage, and that's quite easily visualized by staining for so-called gamma H2AX foci. So every red dot that you see here is a double-strand break where these foci form and then you stain for them. So there's a lot of damage in this organoid. As you can see, there's about 50 organoids or so. All of them have been injected with the bacteria. They're left in overnight. The next day, every organoid is full of double-strand breaks. And the control here, I think a single double-strand break in the entire culture. So we can demonstrate that what was published, that we can recapitulate that PKS E. coli causes double-strand breaks. Now, most of these cells will die. that have the breaks, but the question is, the ones that don't die, will they actually end up with mutations? And here they did the next experiment. This is rather heroic. So rather than just injecting once, they injected about multiple times over 3 months. On the beginning of the week, they would inject the E. coli. At the end of the week, they give antibiotics, because if the E. coli escape from here, they would overgrow the culture, and that would be the end of the experiment. So, and then the next week they inject E. coli again, they have antibiotics. So they go through a large number of these cycles with the bad guy and here the mutant that cannot make the colibactin substance. If we would now, after 3 months, sequence the organoids, we would expect to see many more mutations here than here, because these have been exposed. We would hope to see that these have been exposed to colibactin, but we would have to actually clone out single cells to really see the genome of one cell, because every cell is exposed has a different set of mutations. So we take single cells after 3 months exposure, we grow them up again. If we have enough DNA, we can do a whole genome sequence, and now we can ask, are there more mutations, first of all, in our exposed cells versus the control organoids? And more importantly, if there are more mutations, are they recognizable? Are they of a particular type that we can easily identify? And that turns out to be the case. So first of all, we see many more mutations when we expose the cells to the bad guys, the bad E. coli, versus the negative control that lacks one of those 10 genes, so cannot make colibactin. More mutations, but also the additional mutations are very specific. They always hit this particular sequence that you see here. And what happens is this TA pair is either it's mutated into any other base pair, for instance, CG or GC or AT, or it's deleted. And so in the first case, in the first instance, you would get a missense mutation if it's in a reading frame. And if it's deleted, you get a frame shift. Both are mutations you would see in cancer. And this happens in the context where there is an A at -3. Now, if we now go back to the way colibactin will attack DNA, colibactin, I told you, binds covalently to A residues. So we think it binds covalently to this A here, and then it binds covalently to the A opposite the T, so the A that sits on the other strand. And now you have the interstrand crosslink that I already told you about. This is not compatible with life of the cell, and then the cell desperately tries to get rid of this bit of adduct, and apparently in doing so, it destroys this base pair, so it either removes it or it changes it in the process. Both cases you have mutations that could lead to cancer. Now, these types of mutations were very unique. They had never been seen before, so they should be quite easy to find. This is all very artificial in the lab, but now we could go to a large cohort of patients, 3,500 patients in Amsterdam that had been whole genome sequenced, their cancers. You can see all types of different cancers here. And using that database, we could ask, okay, we have the whole genome sequences from 3,500 cancers. Which of these cancers show signs of the signature that I just showed? So this A at -3, TA at 0, where the TA is changed or is deleted. And lo and behold, we essentially see a significant number of colon cancers, colorectal cancer here, that have this signature. Breast cancers, you never see it. There's the occasional head and neck cancer, where we know that E. coli can also live in the mucosa, and bladder cancer, here is one. These two are, we know now, they were not identified, but are colon cancers. So essentially, we now know that about 15 to 20% of all colon cancers show clear signs of a mutational process that can be started by these bacteria. Now, we then asked these mutations, because so far these mutations were just anywhere in the genome, but do you actually see them changing cancer genes? And that is the case. APC is the gene that's almost always mutated in colon cancers. 80 to 90% of colon cancer cases start with APC mutations. And indeed, we see that in those patients that have mutations that look like they are caused by the bacterium, by PKS E. coli, that actually these patients will have APC mutations that are caused by that particular mechanism. So all of this hints very strongly that PKS E. coli bacteria are dangerous. They are probably carcinogenic, and it's probably a good reason to get rid of them. And that's summarized here. So these bacteria that were known to be genotoxic, they actually induce real mutations. They not only kill cells, and they're quite easily identified. This is their signature here, A at -3, TA at 0, and the TA changes. We see that subsets of colon cancer cases are enriched for mutations by E. coli. We see that the mutations occur in the cancer genes of the colon cancer cases. And there's now many more studies ongoing around the world. This might indicate that it would be a very good idea maybe to detect these bacteria early and to eradicate these bacteria, and particularly in individuals that have a high risk to develop colon cancer. Finally, 2 years ago or so, when the pandemic hit my country, Holland, we went into a lockdown and we were allowed to do COVID research. And of course, we had all these human organs in the lab. We're culturing them, and we asked very simply, and this was done with Mart Lamers and Bart Hagemans in Rotterdam, a real corona lab. So we are the organoid lab, but the virology experts were in Rotterdam. And a friend of mine in Maastricht, Peter Peters, did a lot of electron microscopy also in his lab. We noted that the receptor for the COVID virus is actually highly expressed on gut epithelium. There's a villus here. You've seen villi earlier in my talk. These are the crypts down here, and actually higher than any other site of the human body, including lungs, as far as we can analyze. And we asked maybe not only the lungs. This was March 2020, so we just learned about this disease, a respiratory disease, but we also heard that COVID-19 patients can have gastrointestinal problems, nausea, diarrhea, stomach aches, and that with the first PCR test that became available, it was clear that some people could see the viral RNA in the stool, in the feces of patients. So we asked, does this virus not only attack the lungs, the respiratory tract, but can it also infect the intestinal tract? And that would be important maybe for transmission, but also, for instance, what's being done now. If so, you could follow the virus in the sewers of a city, because if the virus ends up in the stool, you can actually follow how the virus is, how the pandemic is going in a city by taking samples from the sewers and then test them in the lab. Now, that turned out to be the case. It's very easy to infect the human colon and small intestinal organoids, and they make massive amounts of the virus. So in a matter of 2 or 3 days, you get 3 to 4 log increase in viruses. SARS is the original SARS virus from 20 years ago, where the virus again was disappeared by quarantining the patients. For SARS-CoV-2, they didn't work, but they both infect nicely these organoids. Also, they infect lung organoids. I don't show this here. And this is looking at viral RNA. That works well. When we stain, we stain in white for a viral protein, nuclear protein. You can see brightly positive, 1 cell here. These are normal human intestinal cells. If you wait a bit longer, you can see that almost all the cells in the organoid are now infected. What we note is that the ACE2, so the receptors sit on the inside, also in the lungs, so on the apical side. That's where the virus enters. Also, the virus is then secreted when it's amplified back into the lumen. So that's how it rapidly spreads through the organoid, but it will not hit neighboring organoids, as you can see here. And this is, of course, a natural way the virus enters in the lungs or in the intestinal tract. It comes in from the lumen and will hit the apical side of the cells. And that gets to be important in my little story. This was published right rapid. Actually, this whole thing was done in 6 weeks from the start of the experiment to having it published in a journal, which I'd never seen before, with 2 rounds of reviewing, actually. We then embarked on a slightly more complicated story. So now we asked, what are the host genes, so the human genes that the virus needs to propagate itself? And one obvious one is, of course, the receptor ACE2. So there was a control for this approach. Here you can see how nicely the virus infects, SARS-CoV-2 infects human organoids, gut organoids. Here is 2 clones where we knocked out the receptor for the virus. And as you can see, the virus doesn't grow anymore. And SARS, which used the same receptor, so the virus of 20 years ago, also doesn't grow when we knock out ACE2. So this nicely works. MERS is a third member of this subfamily of coronaviruses. It uses a different receptor, and as you see, an ACE2 knockout is happily infected by MERS. So this all fits what we know, and vice versa. If we knock out the receptor from MERS, DPP4, now MERS no longer infects compared to the control, but now SARS-CoV-2 doesn't mind because it actually doesn't use DPP4, it uses ACE2, as I just showed you. So this all shows that this approach works. We then went through a long list of published human genes, and much to our surprise, essentially none of these genes were necessary in our system. And one I want to point out specifically is cathepsin L, was a hit in several in major journals, genome-wide screens, CRISPR screens. Cathepsin L always was in the top, but if we knock it out in purple here, in green is the normal infection rate, in purple it actually grows better rather than that it's blocked. So something is very different between what we are doing here, basically knocking out a gene and then testing the virus, with what was done in those papers. And here's a long list of other genes that essentially shows the same thing. The only gene that we could confirm is TMPRSS2. That did not come out of those genome-wide CRISPR screens, but was found independently. And I'll say more about what that does. We also observed, and you probably have heard a lot about chloroquine as a drug for COVID-19. That actually works in our hands also very well in cell lines that people have been using a lot in virology labs, like Vero E6 cells. So here you see we're growing Vero E6 cells. These are not human cells, they're African green monkey kidney cells, and they look like fibroblasts. You grow them on plastic in 2D, so they're definitely not organoids. And when we now start adding chloroquine, as published, the virus gets into trouble, and the IC50 is about 1 micromolar, and at 10 micromolar of chloroquine, there is no virus detectable at all. So this looks like a great drug for COVID. That's why it has been given to patients early in the pandemic, but it was noted clinically it does nothing. Very surprising because it's so good in culture. But if we expose our organoids, here is untreated in green, 10 micromolar chloroquine in blue, you can see that the virus doesn't get inhibited at all by chloroquine. So very different in 2D cell lines, chloroquine blocks, but in organoids, chloroquine does nothing. So how do we explain this? And then also in the context of what I showed you about the genes. And we published this, but there were some other labs that also came up with the same conclusions. So what happens here on the left, in Vero E6 cells and similar 2D cells, often ACE2 needs to be transfected in because these cells don't normally express the receptor for the virus. If you transfect it in, the virus can bind. It is then endocytosed by the classical endocytosis route and ends up in an endosome. This is, of course, a step that's sensitive to hydroxychloroquine. That's what hydroxychloroquine does. That's why it is a malaria drug. It blocks endocytosis. And genes that came out of those screens that I talked to you about, like cathepsin L, are needed to let the viral genome exit from this viral particle here. So this is what happens in Vero E6 cells, and this explains why hydroxychloroquine works in these cells. However, what happens in real epithelial cells, like we have them in organoids, so the virus comes in through the apical sites. It is now a highly polarized cell. These cells are not really polarized. So on the apical site are ACE2. Virus binds. On the apical site, there's also a transmembrane protease called TMPRSS2. This protease will actually cleave the spike protein. This is the protein on the outside of the virus, but also the protein that makes up the vaccines that hopefully you have been taking. The protease will actually cleave the spike protein. This activates the spike protein. It then forces a fusion of the viral envelope with the host membrane. There's never an endocytic step. The virus genome is then so the RNA is directly injected into the host cell. No endocytosis, and therefore no sensitivity to hydroxychloroquine, and no need for all of these genes that you would need to build endosomes and to let the host RNA genome escape from this endosome. So the lesson from this is probably that for the next pandemic, if another virus shows up, it's probably great to use these types of cells for screens, but also involve something that's much closer to real organs like organoids, and see if what you're finding as a drug really works in a cell that is designed to look much more like a real cell in the lung or in the intestinal tract of a human being. And with that, I think I've been speaking for 45 minutes, so I think I should stop here. I'll stop sharing. I thank you very much for your attention. And I have room for Q&A, I hope.
We're ready for questions.
Thank you for great talk. I enjoy a lot. I have a question about E. coli substance, PKS. Is it toxic to bacteria itself, or they have some special way to protect themselves?
Yeah, that's a very good question. So it's believed that these PKS E. coli make colibactin not to hurt us, but actually to compete with other bacteria in the microbiome. So exactly as you say, so this is believed to kill other bacteria. And in the PKS island, so one of the genes is an enzyme that destroys colibactin. So if the colibactin ends up inside the E. coli cell, it'll be destroyed. So the PKS E. coli are protected against colibactin, whereas all the other bacteria in the microbiome are not. And the fact that they hit human cells is probably unintentional, but is probably quite bad. It probably is a carcinogenic bacterium. Yeah.
Thank you very much.
Hello, thank you for your very interesting talk. As far as I remember, the Omicron variant does not need tempers 2 to enter cells, but it rather uses cathepsins, cathepsin L or B to enter cells. So have you heard of or have you tried any clinical trial to see if chloroquine can really have a clinical effect in this in people infected, especially now that we are immersed in Omicron variants epidemics?
Yeah.
Thank you.
So I would have to ask my virology colleagues. I haven't seen those studies actually. So what we know is that the Omicron is actually better in getting into cells, and it uses both pathways, also in epithelial cells. It would still, so in our hands, the Omicron still enters from the apex by binding to ACE2. We haven't really tested Omicron in the tempers knockouts. That would be actually a very good experiment to do, because we could address this question that you're saying. So if an organoid cell has ACE2, but it doesn't have tempers 2, can the virus still enter? And you would say it might actually go through endocytosis and then use cathepsin L. We did not see, I don't think we saw that they became chloroquine sensitive, though, in the organoids. So it looks that they still can get inside without endocytosis. But it's a good experiment, actually, that you propose. We should probably do this. Thank you.
I have a question to take regarding the organoid formation. I wonder what is the plasticity of the stem cells that you use. For instance, the intestine is a very long structure. I don't know if you transplant in the anterior, posterior parts, they acquire the properties of the position where they are transplanted, or for instance, in the case of the liver, the lobes are different. When you transplant those organoids, they acquire the properties of the position where you put them.
That is a very good question, actually. There's been, so for the gut, quite a lot has been done. And on the small intestinal tract, histologically, it's different in the duodenum and the duodenum and the ileum. The villi gets shorter, there's more pennant cells, and there's different transporters. So you can actually take organoids from different regions of the small intestine. You can also compare small intestine and colon. And for the small intestine, actually, if you keep them in culture, for about a year, you can still see that they were ileum or they were duodenum or they were jejunum by just doing RNA-seq. So they retain their identity and culture over very long periods of time. Colon to small intestine. So if you transplant, there's actually papers published now, if you transplant small intestinal organoids to colon, you create a patch of small intestinal epithelium. So if you transplant, very different from development, because they would be instructed to become colon, but once you take them in an adult, they will remain small intestine. And my postdoc Toshi Sato, who actually set this up originally in my lab, has been a professor in Japan for quite some time. He's actually used this. There is maybe I'm talking a little bit too long, but there is this short bowel syndrome where very young kids, babies, lose most of their small intestine. And essentially, they can no longer take up nutrients. The colon is not very important. It takes up water mostly, but you can live without a colon, but you cannot live without a small intestine. And these kids need parenteral food, IV nutrition. It's a terrible disease and essentially not curable. But what he is developing, which works well, that surgically he takes a bit of the colon. He still does it in animals, but they're going to test this in humans. He takes a part of the colon, inserts it into the skin, but typically done by surgeons. And then he can, so when that all heals, he can now take out the epithelium from the colon loop, put small intestinal organoids in. So now they essentially has a colon with a small intestinal epithelium. And then in a second operation, he puts this in the small intestine. And that works. So he can actually turn a colon into a small intestine. And he's thinking about using this approach, this essentially a surgical approach, for treating these short bowel patients, these little kids that have no small intestine, but they still have a normal sized colon. So he takes part of that colon, turns it into small intestine, puts it back in the intestinal tract. And I think it's a very, very promising therapy for these children. Yeah. So very different from developmental biology. Once the tissue is specified in culture, it is extremely hard to change it. Actually, I don't think we can change it.
There is a question here from Zoom, from Dr. Esperanza Martinez. There are two questions actually. The first one is, have you tested normal good microbiota in good organoids derived from colon?
Yeah, well, this is ongoing. There's not complete gut microbiota, but we are working with several people in the US who study the microbiome. And there's many labs that have made these communities of maybe 10 or 15 species that are stable. So if you culture them over long periods of time, all 15 will be in the mix. And we're now testing to see if we can colonize organoids with a mix, not just with one species that I showed you with the E. coli, but now with something that's more complex and a little closer to the real microbiome. So that's technically doable. We haven't really done any experiments with this yet.
Yeah, the other question is, have papillomavirus been tested in organoids?
Yeah, so we can actually make cervical, so from the cervix, we can make organoids quite easily from a pap brush. So we don't even need a biopsy. So a simple pap brush that's as you know, taken for diagnostics from women. There you can actually grow them as cervical epithelium, and sometimes when it's cervical cancer, you'll see the papillomaviruses in there, and we've been able to infect these cervical. Not so easy. Other viruses are easier, but it does work. Yeah, we haven't done it for skin, for instance.
Another question from Dr. Alberto Darson: can organoids cross the blood-brain barrier?
No, we don't think so. They're much too large. But having said that, there are actually labs, we are not doing this, but there are labs who are building blood-brain barriers in the lab from organoids. And that would be quite interesting to then test, if drugs will or will not go through the blood-brain barrier or design ways of getting through the blood-brain barrier. But organoids would be much too big to travel through an intact blood-brain barrier.
I don't have a question. Is there more questions in the audience?
Just have another Another question just regarding the complications for future use of organoids. You mentioned that you need the material. I wonder if the material is just a thing that needs to be developed to have better organoids. And the other question is regarding the growth factors. You mentioned at the beginning a set of growth factors. And I wonder if all organoids that you have mentioned requires the same factors, or is a complication to get the right recipe for depending on the tissue?
Yeah, both are very relevant questions. So starting with your second question, there is a general recipe, a cocktail that will work a little bit for most tissues, and that consists of 3 components: a potent activator of the Wnt pathway. We used our spondin in the gut example. We now use mostly surrogate Wnt, and this is published by Chris Garcia. It's an antibody-like molecule that works like a Wnt. It binds to the receptors. It's very cheap to make, extremely potent. Wnt is very difficult because it's lipid modified and hard to work with, sticky, expensive, hard to make. So we use the surrogate Wnt nowadays. Second, we typically need a tyrosine kinase receptor ligand, so EGF or FGF or AGF, depending on the tissue. So we need to find out what works best. And third, we need to block BMP and TGF-beta signaling typically. And that can be done with a small molecule or with login that we use a protein. And then every tissue needs to be optimized. But there are now many papers from our lab, but also from many other labs that give the protocols, and many also protocol papers that are very detailed. Say for breast, you will need to add estrogen, for prostate, testosterone, for liver, hepatocyte growth factor, AGF. So the culture conditions can be optimized, and I'm sure that if you pick a particular protocol, it can still be further improved and made a bit cheaper. So all the growth factors are defined. They typically are recombinant proteins or they are small molecules. There's no serum in the medium, so the medium is totally defined. But as you say, the matrigel is a problem. Matrigel is produced by a cancer cell line that is grown inside mouse. So where it secretes, these cells secrete collagen and laminin mostly, and then it's purified from these mice. Clearly not GLP, GMP compatible. It's also quite expensive. And when organoids became very popular in the past few years, there was a worldwide shortage. The companies couldn't make enough of matrigel, so expensive, not enough. Batches to batches are quite different. So it would be fantastic if we could use a synthetic hydrogel, like a packed gel or something like that. It would be much cheaper, much more standardized. But so far, matrigel somehow is magical. So there are some other gels that work a little bit, but matrigel really always works far better. And I should maybe name Minna Bissell, who has worked for 30 or 40 years. She just closed her lab, but for 30 or 40 years on matrigel and growing epithelial cells inside matrigel. So it's basically they like this for some reason best, and many labs are desperate and companies are desperately trying to find something else, but it has not been found, I think.
Okay. There's one more question. Can you have a testicle tubule organoid?
That I don't know, actually. I know that no, I'm not sure that it has ever been tried. That would actually be quite a nice... In actuality, I think we've been able to grow every tissue, every epithelial tissue that we have tried to grow. Sometimes it was a bit harder to get it going, but eventually it always works. So I'm sure this should be possible. Yeah.
Okay. Well, thank you, Hans. It was a really great talk. And really, really thank you for accepting the invitation.
Well, thanks to you all, and have a great continuation of the meeting. Bye-bye.
Bye. Okay, so now ahora pasamos a la siguiente presentación. This is from Dr. Frederick Froment, and Dr. Jean-Louis Charlie is going to introduce.
Hello, Dr. Flamand. Do you hear me?
Can you hear me?
Yes.
Okay, fine.
Thank you for accepting the invitation from Patricia. I will make a rapid presentation of the talk. Dr. Flamma is graduated in the University of Lyon in France, and he made various postdoctoral stages, and now is a group leader in the Institute of Functional Genomics in Lyon, and is the head of a group which works basically on the role of thyroid hormone on brain development and also metabolism. And he will speak about some of these studies. He's a very well-known speaker and a very good scientist. He also works on trying to set up assay for studying the effect of thyroid hormones disruptors. So he has also part of applied investigation. Well, thank you very much, Dr. Flamma, and we'll start.
Okay, I'll share my screen. Okay, you probably can see it. So this is where I am currently talking to you. I'm on the first floor here. And today, so I'll try to share some of our recent results, trying to understand how thyroid hormone exerts a general influence on development. But before going into specific experiment, I would just like to do a general introduction of the topic. Originally, thyroid hormone was described as a hormone that triggers the tadpole metamorphosis in amphibians. So this is a really spectacular process where you will see the tadpoles here within weeks. They will completely change their morphology. They will grow their limbs, first the legs first, and then their arms, and then they will switch from gills to lungs, and their brain will be reprogrammed, and then they will lose their tail, and finally they will escape and live on the ground. So it's an extremely complex mechanism. And what is interesting is that you need coordination, because if the tadpoles, they lose their lungs, they lose their gills before jumping on the floor, of course they will drown. So you need synchronization, and that synchronization is made by a signal. And a long time ago, I think it's actually more than a century ago, someone found the signal triggering frog metamorphosis. This guy was named Friedrich Gudernach, and he had this strange idea of feeding tadpoles with the thyroid gland of a horse. And what happened then, and which is on this slide here, is that the tadpoles fed with thyroid gland, they had a much faster metamorphosis than the one that were fed with the horse thymus. So that experiment showed two things. First thing is that there is a molecule in the thyroid gland that is able to accelerate or trigger metamorphosis. And also that molecule must be some general principle acting in many species, because you could take the horse molecule and it will be acting in frogs. Well, these days we know that this molecule is thyroid hormone. It's called T3 for 3-iodothyronine. So it's derived from tyrosine. You see the iodine here in pink. And the way it works is by binding to nuclear receptor, which are called the TR. There are actually two types of thyroid hormone receptor, TR alpha and TR beta. And these are nuclear receptors, so they are transcription factors. They act as HO dimers with another nuclear receptor, which is called RXR, and they directly act on transcription. So if we look back at tadpole metamorphosis, what happened first is the expression of the TR alpha receptor and the RXR. Then TR beta comes slightly later. And then during metamorphosis, there is a peak of thyroid hormone, first T3, but also T4, which is an inactive precursor of T3. And that there is a clear coincidence with the climax of metamorphosis. So thyroid hormone is not only able to accelerate metamorphosis as an exogenous factor, but it is actually the endogenous factor that triggers frog metamorphosis. So the question is, what is happening in mammals? There's something like a metamorphosis during mammalian development. And if you look at the hormone concentration in babies, you'll see that there is a short period of time, postnatal day 1 and 2, where you have actually a peak in the circulating level of thyroid hormone. And the question is whether that peak of thyroid hormone has a function and whether it also synchronizes on the developmental process. So, of course, there is nothing as spectacular as frog metamorphosis, but maybe there are developmental transitions that are which timing is defined by thyroid hormone. So we're studying that in mice, not in human. And in human, there is also a peak in the circulating level of thyroid hormone. And that peak occurs around day 10 to 15 after birth. And if you think about the transition of the life of a mice between postnatal day 0, so the day where they are born, postnatal day 15, there are major changes. The first change is that the animals, they are originally ectotherms. They cannot maintain their body temperature and they have to hide behind their mother to be at 37 degrees, whereas 2 weeks later, they can run around and they are able to maintain their own body temperature. Second point is that due to the maturation process of the lungs, they are actually hypoxic animals when they are born, whereas they get normoxic 2 weeks later. Well, they are fed only with milk at birth, and then progressively after 2 weeks, they start to eat solid food, so they completely remodel the intestinal epithelium. Finally, the metabolism is also based on glycolysis at the beginning, and it's based for ATP production, whereas because they can breathe a lot of oxygen and have access to other source of food, then they use oxidative phosphorylation for ATP production 2 weeks later. Just one point that I will not discuss today, which is quite interesting, is that the regeneration capacity of the tissue is actually maximum at birth, and then it disappears very quickly as stem cells tend to mature and differentiate. So that transition corresponds to transition in receptor expression, as in frogs. You first have the ubiquitous expression of the T-alpha receptor in all the cells, basically, of the mice. And then TR-beta expression starts in specific tissues at the later stages. So the way we address this question of what the thyroid hormone is doing during the early development of mice is by using mouse genetics. And a long time ago, we modified this THR gene, which is encoding the T-alpha receptor, and we replaced by homologous recombination, the endogenous receptor, with that construct, which is quite a complex construct, and I'll go through that. So in the mice that we are using at the beginning, they are heterozygous. So there is one intact copy of the THR gene, so they produce a T-alpha-1 receptor, and as XR, if it's bound to T3, then it activates transcription. Now, if you cross these mice with a mice that express a Cre recombinase, they will act on this LOXP sequence here. They will do some recombination between the two LOXP sequence. So Cre comes first, and then it makes a deletion and removes the stop cassette that was stopping the transcription. So now the transcription can go through the whole construct, and you start to express new protein. One of them is a tau-LAGZ protein, which is a reporter protein, which actually is not quite useful for our purpose. And the other one is a mutant T-alpha-1 receptor. And that receptor has a specific property of acting as a dominant negative. So we'll antagonize the function of the wild-type receptor. And as a result, you completely block the T3 response of the cells. So just by playing with the expression pattern of the Cre recombinase, you can decide with the system where and when you will block the T3 signaling, and that can be done at any stage in any cell type during development.
So we first did that by crossing these mice with mice that express a Cre recombinase at a very early stage of development, so that we can get recombination during early embryogenesis, and we express a mutation in all the tissue of the mice. What you get then is a mice that looks very much like a hypothyroid mice, a mice that is devoid of thyroid hormone. So you see this mice was born with a mutation. That one is a little mate, same age, they are 2 weeks old. And this one is much smaller than the other one, just like a hypothyroid mice. And they have a very small spleen, for example, and they also have a major retardation in bone ossification. And finally, these mice, they will die within 3 weeks after birth. So there is really something really important that is antagonized by this mutation of the TR-alpha receptor. And this is what we really want to understand. And so we started to play with different Cre recombinase. And over the years, this is a full collection of all the Cre that have been used in our lab and in other labs under collaborative project to block thyroid hormone signaling, either in the muscle or in the liver or the bone, the cartilage, thyroid gland, etc., etc. What turned out is that we get lethality and death after birth in 2 cases when we express a mutation in all the brain cells. So you will get exactly the same type of lethality 2 weeks afterwards. Or if you restrict the expression to a specific category of neurons, which are called the GABAergic neurons. So you should know that in the brain, there are 2 types of neurons to really simplify things, the excitatory neurons and the inhibitory neurons. And the main neurotransmitter of inhibitory neurons is a GABA. So the GABAergic neurons are the main inhibitory neurons of the brain. So there was something very important going on with thyroid hormone receptor in the GABAergic neurons. So why do they die? It took us a long time to understand that until we decided to put a video camera on top of the cages. And you will see here, this is the mother. These are the pups. They are in a nest with cotton. And this is one pup with a mutation. And look at what's happening in the cage. So that pup is in the cage here. And the mother is trying to catch it, but it's really excited. And actually what's experiencing are epileptic seizures. So the mother wants to bring it back to the nest, but each time she catches it, then it triggers epileptic seizures. So you'll see another one here coming. So typical epileptic seizures. And so what's happening and why the mice died, there are actually 2 causes to their death. The first point is sometimes epilepsy itself can kill the mice. But we think that more frequently what's happening is that in the absence of maternal care, they don't get any food and then they die. So if you generate a mutation of the TR-alpha receptor in the GABAergic neurons, you will have epileptic seizures. And now we have to understand exactly what is going on at the cell level. So the GABAergic neurons, you see them in green here on this slide, because it's a GFP GAT2 construct. Well, they are all over the brain. And over the recent years, we have been focusing on 3 brain areas. Striatum first, because it's really filled with GABAergic neurons. So for biochemistry, it's much easier than other brain areas. Cerebellum is another model that we have been working on just because there is a lot of literature, classical literature on the function of thyroid hormone during cerebellum development, but I will not discuss that today. And finally, the cortex, of course, is a place where you have all the cognitive functions and where a small fraction of the cells are GABAergic neurons. So today I will just focus on the cortex. So a little bit of context on the visual cortex and a very important concept in neuroscience, which is called the critical period. What you see here are 2 electrophysiologists from Harvard Medical School who did a really fascinating experiment in the 1970s. So this scheme shows the visual track, the human visual track, where you see the eyes here, the project from the left and right part of the cortex. That part of the left eye project to the right hemisphere, whereas that part and that part goes to the left hemisphere, so that the circuit, the classical circuit of the visual system in the human brain. And what really matters is that depending on where you go, where the light comes, it will excite the neurons on one side of the brain. Now, the experiment that they did is just to take a small cat at the 4th week of the postnatal week of its life, and close the eye of the cat just for 1 week. What's happening then is that here what is occurring, what you can record, the electrical activity in the cortex of a normal control cat. If you the light comes from the left eye, you will excite the neurons on the right side of the brain, and vice versa. Whereas if you close the eye on one side, then you completely lose the capacity to excite the neuronal activity on one hemisphere. So what this experiment is telling is that first you need some excitation and some training to elaborate the neuronal circuit in the cortex. And second, that takes place during what they call the critical period. And that has to be in the cat, the 4th postnatal week. If there is no light coming and so no excitation of the cortex during that period, then the cortex will never learn how to process the light. Okay, so what about the mice? There is also a critical period in mice. And this is the current knowledge on the maturation of the circuits in the neuronal circuits in the cortex. Well, at birth, you still have the two major neurotransmitters in the cortex, glutamate for excitation and GABA for inhibition. The onset of the excitatory activity, so the electrical activity that you can record in the excitatory neuron, is very early, postnatal day 7. Then at postnatal day 14, the mice that are born with the closed eyes, they start to open their eye, and that start to, the excitation start to flow into the cortex. Then only after that comes the inhibitory activity of the GABAergic neurons. They have made synapses with the excitatory neurons before that. And after, let's say, 1 week, there is a balance between inhibition and excitation. And that period is a critical period of high synaptic plasticity. It is where the cortex trains to process the light signal. So basically what's going on in our mice is that they have a defect in inhibitory activity, and so there is only excitation. And when excitation causes excitation, what you have is epileptic seizures. So clearly what we suspect is that when you put a T-alpha mutation in the GABAergic neurons, they cannot be matured, they don't have the function, their inhibitory function, and they cannot counterbalance the excitation caused by the excitatory neurons, and that generates epileptic seizures. Okay, in more details, we did some, this is how the neural circuits work. Well, glutamatergic neurons are called the pyramidal neurons, doesn't matter here, but the input comes from outside of the cortex, excite this pyramidal neurons, but then, for example, the basket cells here that express parvalbumin, they exert some kind of feedback because the excitatory neurons will activate the activity of these inhibitory neurons, and that inhibitory neurons will inhibit the neurons. So there is a negative feedback loop, electrical negative feedback loop, based on basket cells here. These cells here, they have also a similar function, except that they don't connect at the same place on the excitatory neurons, and they express somatostatin. And finally, very important point, during the critical period where you really need to set the wiring and all the correct synapses for the correct processing of the signal, what's going on in this basket cells is the elaboration of a specific type of extracellular matrix, which is called the perineuronal net. And it has been shown that that specific extracellular matrix is very important to stabilize the synapses around the basket cells and to ensure that the negative feedback will work properly. Okay, so let's go back to the experiment. Just one point is that I will be discussing these GABAergic neurons of the cortex a lot these days. but and talking about these markers. So there are actually many ways. I just discussed the basket cells, for example. Basket cells is based on their morphology, but also you can characterize them by immunostaining because they express this parvalbumin antibody, antigen. The somatostatin positive cells are different categories of cells, the Martinot T cells and so on. So you have all this classification based on immunostaining and then on the morphology. And nowadays, people do single-cell RNA-seq, and then they redefine all the cell type based on gene expression. And they suspect that there are actually many more categories. So it's not clear at this point which cell is which one and so on, and whether they are actually really different type of cells. But clearly, I will stay today with the parvalbumin cells, somatostatin cells, even though we know that behind this denomination, there are different cell types. Sorry about this. Okay, so we did some immunostaining on these mutant mice. First point is that if we use a tomato staining, which is based on the Quilox system again, so we turn on the expression of the red tomato protein in all the GABAergic neurons. You see no change in the density in the cortex for these cells. So we don't lose neurons. All the neurons are present, but they don't express necessarily the markers of terminal differentiation. In particular, you lose the parvalbumin staining. This is control mice here that have no mutation in T-alpha. These are the ones with a mutation in T-alpha. You see very few staining with parvalbumin. So you lose the capacity of the parvalbumin cells to terminally differentiate. Whereas if you use somatostatin immunostaining, you don't see any change. So more precisely now that we know that the mutation of T-alpha has a specific effect on the maturation of the parvalbumin expressing cells, so basically the basket cells. So what can we do to understand what's going on into these cells now? There are only a small fraction of the cell population, maybe 5% of the cells in the cortex. So we try to do whole cortex RNA analysis, but of course, it's not really successful with that. So we use this technology of cell sorting, actually nuclei sorting. So we just do reporter mice that express a green protein into the nuclei of the parvalbumin neurons, I mean the GABAergic neurons. Then we combine that with a mutation of T-alpha, or we combine that with hypothyroidism that you can generate with a drug, or we take the hypothyroid mice and we treat them with thyroid hormone. In all these mouse models, what we do is nuclei sorting using a cell sorter to sort out all the nuclei from the GABAergic neurons, so a small fraction of the population. And then we use RNA-seq and deep sequencing to analyze gene expression specifically in that subpopulation. So we did that, and we got this heat map with many, many genes which are expression is changed. So these are the control mice. In blue, this is low expression. In red, it's high expression. And you see that many genes, they are downregulated here in the mutant mice and also in the hypothyroid mice. So it's the same. I mean, whether you express dominant negative T-alpha mutation or you make the mice hypothyroid, you get the same consequences that you downregulate target genes of the receptor. Now, if you restore the level of thyroid hormone by giving some thyroid hormone to the hypothyroid mice for only 24 hours, then you restore the expression of these genes. So these are genes that are sensitive to thyroid hormone or to the mutation of TR, even if the mutation is only in the GABAergic cells. However, you see that there is an equal number of genes that are just behaved in the opposite direction. Their expression is higher in the mutant or in the hypothyroid mice. And so these genes cannot be the direct target genes of the receptor because we know that the receptor is a transcription activator. So transcription repression must be some indirect effect of having the mutation or being hypothyroid for a long time. So next step for us is among these 400 genes, can we identify the true target genes of the receptor? Meaning that can we really identify the molecular mechanism that explain how the mutation of the T-alpha receptor blocks the differentiation of the parvalbumin population? So just one point we extracted from the previous data. So it's just a something that came out of this same result. The expression of the ion channels, potassium channels, and calcium channels that have been reported in this paper from Okati in 2009 to be good markers of parvalbumin cell maturation. So, and I mean the GABAergic population, but mainly the parvalbumin population. And so what they found is that some of these ion, I mean, there is a real reorganization of the ion channels in these neurons after birth. Some genes will come up with time, some will come down with time. So I just put this in red, the genes that are normally should be coming up with time, and in blue, the one that should normally come down with time. And you see that compared to the control mice, the mice with the T-alpha mutation, basically they have less expression of the gene that should be increasing with time, and higher expression of the gene that should be going down with time. So basically, they are retarded in their differentiation program. It's the same in a hypothyroid mice, I mean, more or less the same, but you do not recover that when you treat the mice with thyroid hormone. So there is some irreversible damage that you cannot restore with a short treatment of thyroid hormone in the repertoire of ion channels. So that is close to the function, the electrical function of the GABAergic neurons, but this is not a molecular mechanism explaining how the T-alpha mutation prevents the maturation of these neurons. Now, if we focused on those genes that just behave like should behave, the thyroid hormone receptor target genes, meaning they should be down in the mutant mice, they should be down in the hypothyroid mice, and they should be restored by thyroid hormone treatment. And we do what is called gene set enrichment analysis. We'll find that many of these genes encode component of the extracellular matrix, like the collagens here, or specific component for the perineuronal net, which is this very specific extracellular matrix of the basket cells, or they are involved in making the glycan that are present in this extracellular matrix. So many markers suggesting that there is a defect in the capacity of the parvalbumin neurons to make the perineuronal net. Well, of course, you don't need to do RNA-seq to do that. You can just look by histology. And there is actually a lectin called WFA that can stain the perineuronal net around the parvalbumin neurons. So you see in the control mice here, in red, this is parvalbumin staining. In green, this is the perineuronal net staining. That's a normal situation 2 weeks after birth in some part of the cortex. And this is what's happening in the mutant mice. First, you don't see parvalbumin, or very few parvalbumin, and the perineuronal net staining is also very, very reduced. So there is a major defect in the maturation of the parvalbumin neurons. And one thing that they cannot do is to generate this perineuronal net, which has certainly a major functional consequence, because we know that the perineuronal net is important to stabilize the synapses and to ensure a proper feedback regulation of excitatory neurons. So now the question is this irreversible, and what does that happen? So we switch to another model. We again use the Krylov system to activate the mutation of the T-alpha, but this time we used a Kry that is fused to the tamoxifen inducible part because it's fused to the estrogen receptor. So the recombination will take place in the mice only if we give them tamoxifen. And we started to use this system by giving tamoxifen to the mice 7 days after birth. So if the hormone, thyroid hormone, is required for parvalbumin neuron maturation during the first postnatal week only, then we should not see much of a consequence of giving the tamoxifen. If it's later, then we should see it. And the answer is on this slide. This is parvalbumin staining again. This is a quantification of the slide. This is all the GABAergic population again. No change in the overall density in GABAergic neurons, but the parvalbumin maturation does not take place. So the thyroid hormone is doing something after postnatal day 7, which is required for the maturation of the parvalbumin neurons. And now, is it only transient, or is it something that is needs to be present all the time, meaning if we wait long enough, if we wait for the cell to express parvalbumin, do we need the hormone to maintain this function, or is it okay? I mean, once they pass the maturation process, can they still do they still need the thyroid hormone? So we switch again to another model. This time we use another Cree, which is actually based on the parvalbumin expressing gene. So the cell will express the Cree recombinase only once they express the parvalbumin. So basically after maturation. So we look at these mice, and for the moment it seems that they don't have much of a phenotype. At least the parvalbumin staining is normal. Now, so it tells us that what the hormone is doing is basically between day 7 after birth and day 15 after birth. However, we look at adult mice with this model, because these mice, they don't have epileptic seizures, so we can look at the adult. We sorted out nuclei from the parvalbumin cells, these cells, and we look at gene expression in the nuclei by RNA-seq. And what we found is there are actually changes in gene expression in these mice. So, for example, this hairless gene here is a well-known target gene of the receptor. SEMA7A is also has been reported to be a target gene of the T-alpha receptor. But for the others, we're not sure yet, but it seems what there is something permanent, a permanent problem. You need the receptor, the functional receptor, for having perfectly fine parvalbumin neurons. So it's not clear at this point for us what is the function of the receptor in the adult, I mean, after the maturation of the parvalbumin neurons. But with this model now, where we have expression only in the mature parvalbumin neurons, we'll look at the mouse behavior and see whether there is something wrong with expressing a T-alpha mutation at adult stage in these neurons only. Okay, so just to summarize this part, and then I'll switch gears. Well, I told you that thyroid hormone in the tadpole is acting as a signal to synchronize very important developmental event. I think we have good evidence that this is also true in the cortex. Well, between day 7 and day 21, there is a maturation of all these neuronal networks. And this is really dependent on thyroid hormone. If we express a mutation, or if we reduce the level of thyroid hormone, we have less expression of parvalbumin, so we have impaired maturation of the parvalbumin expressing GABAergic neurons. The number of synapses, I didn't show you that, but some people show that the synapse formation is also dependent on thyroid hormone, the elaboration of the perineuronal net, and that switch in the ion channel repertoire is also very important. And so the somatostatin neurons, they appear at an early stage after birth, and they don't need thyroid hormone for the differentiation. But the parvalbumin expressing neurons, meaning mainly the basket cells of the cortex, they really need the thyroid hormone for the proper maturation at the proper time, at the right time. And this is dependent on thyroid hormone. And finally, if you don't have that, you don't have the proper balance between excitation and inhibition, and you may have epileptic seizures. Okay, so that's it for the basic science on thyroid hormone and brain development. Now I would like to switch gears and just to tell you about the disease, just in case if there are clinicians in the audience, because there is actually a genetic disease due to T-alpha mutation in human. This is actually quite a recent discovery. The first patient was reported in 2013 by Chris Chatterjee in London, or in Cambridge, I'm sorry, in England. And that girl came with a short stature, and she was giving growth hormone and was resistant to growth hormone. And she had also some bone malformation and problem with the hips and so on. And the skull was also really, the suture were different and so on. So it was really a strange case, and they didn't understand at all the pathology, and they decided to do exome sequencing. And they found a T-alpha mutation. And then Chris Chatterjee, who knew very well the consequence of T-beta mutation in humans, was really surprised that he didn't understand that it was actually a patient with a T-alpha mutation, because that was actually predictable. Okay, now these days, it's a very rare disease, but there are 40 patients and I think 30 families with a T-alpha mutation. This is a 3D structure of the 3' end of the nuclear receptor T-alpha. This is a thyroid hormone fitting in the ligand binding pocket. This is another representation of the same structure. And what is really important is this terminal helix is here. Helix 12 here is required for the interaction with some class of protein are called transcription co-activators. If that protein is not present, then the receptor cannot activate transcription. But not only that, but it also recruit transcription co-repressors. So instead of acting as a transcription repressor that is switching to transcription activation in the presence of hormone, it becomes a permanent repressor. And that explains the dominant negative effect of this mutation. So these are all these mutations in the patients. And surprisingly, some patients are really severely ill, just like very severe congenital hypothyroidism. And some other patients are just fine. And some have really strange symptoms. So we thought that we should use a mouse model to model the genetic disease. And in fact, the mice that we've shown before is actually a first model of the genetic disease with epileptic seizures that have been reported in the human patients also. So for basic science, I think if you like basic science, you should tell the story. We made a model for a disease that was not discovered yet. But when the disease was discovered, then we immediately added a model. Now, we didn't have a precise model for precise cases, and we wanted to do better. And at that time came the CRISPR-Cas9 technology that enables us to speed up a lot the generation of new mutation in mice. So basically, the way we do it these days is just so simple that it's embarrassing. What we do is just with electroporate oocyte that we place in this chamber, and then we introduce into the medium here what are called ribonucleoprotein, where you get this CRISPR, the Cas9 nuclease, which is a recombinant protein, together with a guide RNA. And if you know the theory behind genome editing, you know that if the first 20 nucleotide of the RNA just hit just hybridize a very specific site of the genome, then the Cas9 will cut that genome exactly there. And either you generate insertion or deletion, or if you add on top of it, on top of the Cas9 and the guide RNA, if you add a single-stranded DNA, which is called a single-stranded oligodonor DNA, then you can use that as a template, and you can even introduce point mutation and do site-directed mutagenesis in the mouse genome. So we did that early and generate frameshift mutation just like they were present in the human patient. So this is again how CRISPR-Cas9 works. This is a sequence of THRA exactly next to the helix 12. This is a sequence that we are targeting with a guide RNA, and we know that also, for those who know the system, you need these 2 Gs next to it, which is called the PAM motif. And so this is where the Cas9 will cut the DNA. Well, generating a cut does not generate a mutation because the cell will repair the cut by a process that is called non-homologous end joining. And then they repair the DNA, but then Cas9 will cut again until the cell makes a mistake during the repair process and generates small insertion or deletion. So these small indels are actually very equivalent to what is observed in patients. And so if you do that in mice, you generate random frame shifts. And that's what we did immediately, and that was very efficient. So the second possibility, we wanted to copy exactly a mutation that was present in one patient. There was a missense mutation this time. So you still have this non-homologous end joining process going on, and you generate small indels also. But now you introduce this single-stranded oligo as a template, and this is a sequence of the oligo. And sometimes the cells will use another way to repair DNA. Instead of using non-homologous end joining, it will use homology direct repair, and it will be fooled by this oligo because instead of using the intact copy of the gene for repair, it will use the oligo, and then now you dictate exactly the nucleotide change that you want to do in mice. So we did both. And that's what exactly what the mutation that we did by introducing this change in the coding sequence of THR. So we did both, and this was a situation a few years ago. Now it's slightly outdated. So there were the human mutation that were the first one to be reported. You see this one is a missense mutation, that one also. These are frame shift mutations. And these were the mouse models that were present in the literature. The one that we did was L400R, the one that I showed before. Very surprisingly, someone in Sweden did a mutation with a C here, which is exactly the same that was found in one patient 10 years later. And then there was one frame shift, but it was not exactly the same that in the human. And so these are the ones that we generated with CRISPR-Cas9. So when you do frame shift, there are two possibilities because there are three reading frames. So the first possibility is one nucleotide deletion or two nucleotide insertion, and you generate that reading frame ending with an L, and that actually shortens the protein, just like in the patient. You should look at the similarity between that frame shift and the one that has been reported in the last patient here. Or you do the other frame shift, and then you get a different reading frame. And in the case of THI, that enlarges the size of the protein. So you get a bigger protein. And that has not been reported in patients. So we were asking, would the mice with that type of mutation, would they be like the others? Or would they have a stronger phenotype or weaker phenotype or different phenotype? So we look at the phenotype of these 5 models, the 2 with a short receptor frame shift, the one with a frame shift but with a long receptor, and the one that we copied from the first patient here, which was a very strange patient with very specific symptoms. So these are the tail vertebra of these mice 2 weeks after birth. You see in the control, you see what is called secondary ossification. On the both sides of the bone, you see this ossification that's a normal situation. Now, if you do a mutation in T-alpha, 1 allele, all the mice are heterozygous, just like the patient. There is retarded ossification in bone growth, and you see no secondary ossification with these 2 mutations that are short frame shift. Whereas if you look at the long frame shift, the one that extends the protein, clearly you have a weaker phenotype, but you have a slight delay in ossification if you compare to the control. So obviously on that side, these long proteins, they don't have such a strong dominant negative effect, and the phenotype is weaker. And finally, that missense mutation that was reported in the patient didn't have any effect, not only on bone ossification, but also on bone, there's no bone malformation and so on. And the conclusion basically here is that the clinician described many traits and many symptoms that are unrelated to the T-alpha mutation present in the patient. Okay, then we move to gene expression analysis in the heart, in the cerebellum, and these are the well-known target genes of thyroid hormone, HTN2 in the heart, HRLS in the cerebellum, TLF9 also. And basically, when you have a frame shift, you have a much lower expression of this target gene of the receptor, both in heart and cerebellum. But the effect of the mutation is much stronger if you have shortened the receptor, so you lose helix 12, but you also reduce the size of the protein compared to the long frame shift, the one that extends the receptor. So the dominant negative effect of in the mice seems to be stronger with a short receptor. Now we'll switch completely to in vitro work, and we look at dominant negative effect. So what we did is just transfect cells with wild-type receptor, mutant receptor, and we put thyroid hormone on top of that. And let's say we have the wild-type receptor and we increase the amount of receptor that we transfect into the cells, and together with thyroid hormone, and this is a reporter with the DR4 elements, I mean the binding site for TRXR, and we just look at luciferase activity, while more receptor, more luciferase. Now, if instead of adding the wild type receptor, you add the mutant receptor, you start to see the dominant negative effect. You decrease a lot luciferase activity. And again, if you have a receptor with a short frame shift, I mean the shorter protein with a frame shift, the first type of frame shift, you have a strong dominant negative activity, just like it's the case in the patient. Whereas if you extend the receptor, you basically see no dominant negative effect in this effect. And again, that point mutation doesn't have any effect on the transactivation. So a very clear correlation between this in vitro observation of the dominant negative effect and the phenotype of this mice, strong phenotype, strong dominant negative effect. Finally, we look at the interaction between the co-activator, which is called NCOR, and the T-alpha receptor. So it's a slightly complex experiment. It's an in vitro experiment again. It's called a two-hybrid experiment where we fuse the VP16 activator to the receptor. So it should act as a permanent activator of transcription unless it interacts with the NCOR receptor. And then to activate, you need to bind to DNA because we have only the ligand binding domain here. And to bind DNA, we use a GAL4 binding site. So what you measure here by measuring the luciferase activity is the interaction between the ligand binding domain of the receptor and the co-repressor. And so if there is a strong interaction, which should be the case in the absence of hormone, then you have a lot of luciferase activity. Whereas if you add the hormone, you should reduce interaction, you should reduce the luciferase activity. This is exactly what's going on with wild type receptor. I mean, in the absence of thyroid hormone, you see the interaction between the co-repressor and T-alpha, and so you have luciferase activity. But as soon as you add thyroid hormone at growing concentration, you see no activity. If you do that with a mutant receptor, again, with a short receptor, the first type of frame shift, you see a very strong and permanent and thyroid hormone insensitive interaction with the co-repressor. If you have extended the size of the receptor, there is some kind of hindrance, and then you will get less interaction with the co-repressor. And again, with this point mutation that is found in a patient, you see basically no effect of the mutation. And so, no dominant negative. Okay, so I would like to conclude, not only on that part, but on the presentation. That genetic disease is called RTH-alpha for resistance to thyroid hormone due to T-alpha mutation. Maybe you have seen patients if you are clinicians, so please let me know because they are very difficult to recognize because they don't go to endocrinology because they don't have normal level of thyroid hormone, but they look very much like hypothyroid patient. They have mental retardation, they have epileptic seizures, they have small stature, they are just like hypothyroid patient. This mutation, they may not be so rare, but the diagnostic is really difficult. The mutation turned the T-alpha-1 receptor into a dominant negative. And this is due to the permanent interaction between the receptor and the co-repressor and its inability to recruit co-activators. So the interaction between co-repressor is not balanced with interaction with co-activators. So disease severity, I showed you that with the mouse model correlates with the strength of the interaction. And finally, it just illustrates the fact that now that we have CRISPR-Cas9 working, genome editing can be used to generate avatar models. So you have one patient with specific symptoms, you can generate the mouse model within few months, and you can study the phenotype of this patient. With that, I think I will end by thanking the group who did the experiment. Especially, I would like to thank Suzy Marcosian here. She did the genome editing. Romain Guyot is the one who did the RNA-seq. I didn't talk about the metabolic part that was done by Karine Gauthier, and the project on the cortex is led by Sabine Richard, which is on the left in this picture. So I'll stop here and take any question if you have them.
Well, thank you very much, Dr. Flamand. It was a very nice talk. Questions? I will start with one. I suppose that what we have seen on the GABAergic neurons may be generalizable to other neuronal types. Do you know? Do you think it's the case, or?
No, I don't think it's the case. And it's really strange because that's what we started originally. We saw that we would generate models where we use the mutation excitatory neurons and inhibitory neurons and check that. We did that in a cerebellum, and we found that the terminal differentiation of the excitatory neurons of the cerebellum, which are called granule cells, it's really insensitive to T-alpha mutation. Perhaps they express less receptor than the others, but they don't need the hormone for the maturation. If you look at hypothyroid mice, you might see differences in altered maturation for basically all the cell types. But what we found is that the primary defect comes from the GABAergic lineage. And under the stimulation of thyroid hormone, this is very clear in cerebellum. Some cell will generate some neurotrophic factors and growth factors. And indirectly, this is important for the maturation of the other neurons. But the literature says that, for example, the granule cells, they really need a thyroid hormone for their terminal differentiation. This is not a direct effect. And the same thing with the oligodendrocyte lineage. There is many papers describing that they need the hormone for their differentiation. And this is, again, an indirect effect of the hormone. So there is a specific sensitivity of the GABAergic lineage to thyroid hormone for a reason that I don't understand. Maybe they express more receptor. I'm not sure, because we cannot really quantify the proteins. We don't have good antibodies for that.
You can directly read the question in
Yes, I see it. So the question is, does the ability of the T-alpha mutation to bind coactivators occur in vivo? Could it then inhibit the transcriptional effect of many other transcription factors? So I should have give a slide on the mechanism of action, the balance between coactivators and co-repressors. What really is important is that none of the interacting proteins that interact with the TR, either in the absence of hormone, which are the co-repressor, or in the presence of hormone, which are the co-activators, none of them are really specific for TR. They are used by the other nuclear receptor. They are used for general transcription mechanism. Basically, they are histone acetyltransferase, histone methylase. This is a histone code story. So now, the ability, there are possibilities for crosstalks. So you can imagine, for example, that if you trap the co-repressors with a mutation of T-alpha, you will have indirect effect on other signaling pathway. We have no evidence that this is taking place. And I think the reason for that is that the TR in the number of molecules per nuclei is really small. So the TR use proteins that all the other nuclear receptor and many other transcription factors use to work. But I don't think there is competition or, there have been papers reporting in vitro mechanism for crosstalks, but I'm not sure. Clearly, the interaction takes place in vivo, for sure. But for example, NCOR is not the only one. And we tried to cross the T-alpha-1 mutation with the NCOR knockout, expecting that we will release the repression and the mice that would survive. That didn't happen. So there are other co-repressors for sure. So it's a complex sort of story, the story of co-activators and co-repressors. And the next question is, TR-beta is essential for development at another stage. While TR-beta comes later, the patient with TR-beta mutation, they have mild cognitive problems. They have attention deficits. They are not really good at school. They have low IQ, few points below. So TR-beta is also involved, but it's not as essential as T-alpha.
I have a question here regarding the first part of your talk. I wonder if you have checked the levels of GABA. I mean, if levels of GABA are affected, because thyroid hormones and GABA regulate myelination. And my question is, have you checked myelination, if it's affected? Because
yes.
Probably the is also related to that.
So it's a very interesting question. We did not measure the neurotransmitters. We plan to do some metabolomics at some point. I thought that there should be an effect because GABA comes really early, but we haven't done it. Now about oligodendrocyte lineage, we know that there is a defect in the myelination if you express the T-alpha mutation. I'm sorry, there is no effect on myelination if you express the T-alpha mutation in the oligodendrocyte lineage, but there is an effect of hypothyroidism. So that leaves open many possibilities for indirect mechanism. Some people have been involving neurotrophin-3, BDNF, all kind of neurotrophic factors. Sonic hedgehog could be another one. All these are made under thyroid hormone stimulation by other cell types. But GABA itself, I didn't know it was so important for oligodendrocyte differentiation, but it's a possibility, yes.
Thank you.
I have a question about the first part of your talk. This T3 function that you discover in GABAergic neurons of mice is something similar demonstrated for other vertebrates that undergo metamorphosis?
I'm not sure. I know that they are seeing, I mean, the frog metamorphosis, for example, there is brain rewiring, but I'm not sure that the neurons that are sensitive to thyroid hormone are GABAergic. I don't know, actually. I don't know about the GABAergic system in non-mammalian systems. I should check that. Thank you.
And another question you also showed something about the channels, the chloride channels. Are they defective or affected in the function? Or?
Okay, so we know that the basket cells, they are called by electrophysiologists fast-spiking interneurons. So they need a lot of energy and a lot of ATP because they fire a lot. And in another model, we haven't done that yet, but we plan to do that actually. But some electrophysiologists showed that the firing of the fast-spiking interneurons, so which are basically the parvalbumin neurons that we're looking at, the firing is at the correct intensity, but it stops very often. So they are short bursts, and then they stop, and then the short burst again. So there is something wrong with the electrophysiology of the parvalbumin neurons. So that could be a consequence of the change in the repertoire of channels, yes. But, you know, electrophysiology is something really complex. So between the change in ion channels and the actual recording of electrophysiology, I'm not sure that I can understand that myself, at least. I think one possibility would be that there is a shortage of ATP, actually, only in these neurons, so that they cannot fire all the time. They have to refill their mitochondria with ATP. Before being able to fire again. That would be a unifying hypothesis because we know that thyroid hormone is very important for ATP production and mitochondrial function, but I have no evidence.
Okay, there's still like possibility. There's one more question in your...
yes, I see that. Okay, do you believe, as the literature still points, that TR receptor with no hormone bound are sitting on the DNA when chromatin is compacted? I can think of squelching effect binding to co-repressor, but bound to DNA. Okay, so the answer is yes. If we do ChIP-seq analysis, we have done ChIP-seq analysis. We haven't done that even in the GABAergic neuron of the striatum. I didn't show that today. You would see chromatin binding, both in the absence and the presence of hormone. But this is not permanent, and sitting on the DNA is not a correct image, because if you read the literature, and notably the microscopy done by Brian Haugen at NIH, it shows that the interaction of nuclear receptor in general and TR in particular is highly dynamic. So the receptor, they keep touching DNA, then come back and forth all the time. And the resilience time is really, really short. It's less than 1 second. So there is very, very quick turnover in the dynamic exchanges. So competition between nuclear receptor then becomes difficult to understand. And also the dominant negative effect also becomes difficult to understand, because my original idea was that, well, the receptor is sitting on DNA with a co-repressor bound to it, but clearly there is a turnover. Apparently, the residence time is longer when there is a co-repressor than there is a co-activator. That may explain the dominant negative effect. But crosstalks are possible because some nuclear receptor, for example, the LXR, the RXR, also bind to the same response element. And so it's possible that, generally speaking, some genes are using the same response element to respond to different hormones. This is not so clear at this point. I don't have a good example of that, but it's possible, yes.
Okay. Thank you very much, Dr. Flamand. We enjoy it very much.
Thank you. So thank you for the invitation, and enjoy the rest of the meeting. Thank you. Bye-bye.
Thank you. You're welcome. Thank you. Entonces, ahora vamos a escuchar la conferencia del doctor Brinstein. El doctor Darson nos va a hacer la presentación. Doctor Darson, ¿puede abrir su micrófono para que lo escuchemos?
Hola, ¿qué tal? Buenas tardes. Realmente, ¿sí me escuchan?
Sí.
Hola, Sergio.
¿Qué tal, Beto?
Muchísimas gracias por aceptar dar una segunda plática. Sergio nos dio hace unas cuantas semanas una plática del instituto y accedió muy amablemente a dar otra, que es una muestra de su extraordinaria productividad. Sergio y yo fuimos compañeros del doctorado, él en el Departamento de Fisiología y yo en Bioquímica en el Centro de Estudios Avanzados. Después él y su esposa Mira, que también es conocida amiga del instituto, se fueron a hacer un postdoc a Canadá, al Sick Kids Hospital. Ahí fue postdoc y luego fellow. Después hizo una estancia de 1 año en Zúrich con el doctor Semenza y regresó a Canadá, en donde en el Sick Kids se volvió el jefe del departamento del programa de biología celular casi durante 20 años. Ha tenido todas las distinciones que se pueden obtener en Canadá, incluyendo algo que es nuestro equivalente del Premio Nacional. Ha sido fellow del Howard Hughes, está en múltiples y las más importantes comités editoriales de revistas y tiene publicaciones, más de 550 publicaciones, incluyendo Science, Nature, Physiological Reviews. Es realmente un orgullo y en cierto sentido una pena de nuestros destacados mexicanos que salen del país porque no les ofrecemos las condiciones adecuadas para trabajar. Muchísimas gracias por darnos esta plática.
Gracias, Beto, por la presentación. Y quiero recordarle a la audiencia que mi idioma principal es el español y que con todo gusto me pueden hacer las preguntas durante o después de mi plática en español, pero la plática misma la voy a hacer en inglés porque es el idioma de la ciencia y porque las diapositivas están presentadas todas en inglés, pero no tenga ningún empacho en detenerme y hacer preguntas si hay algo que no está claro. So what I want to discuss today is how we sense blood flow in our arteries. When the blood is pumping from our heart through our arteries, the shear produced by the flow is sensed by the endothelial cells, which is the most intimate layer of the blood vessels. And sensing the flow is not at all trivial. In fact, it has extremely important physiological consequences. Two of them are particularly noteworthy. One of them is that the endothelial cells align in the direction of the flow. The second one, which you're probably more familiar with, is the production of nitric oxide. Nitric oxide, as you all probably know, is extremely important in the relaxation of smooth muscle and the establishment of blood pressure, but also it prevents the adherence of platelets and neutrophils and macrophages, which reduces clotting and autoimmune responses. And these two important responses are better illustrated in this second slide. And I hope you can see my pointer. What is shown here are images of endothelial cells grown in culture under static conditions, meaning no flow is applied. And in that case, the cells are oriented randomly and they have polygonal shape. If, however, you apply flow, as is the case in a normal blood vessel, the cells become very elongated and they align along the direction of the shear flow that you apply. The right panel shows the nitric oxide production. And it's important to point out that the response to nitric oxide is originated by an influx of calcium, and I'll get back to that a little bit later, and that the increase in cytosolic calcium induces binding of calmodulin to eNOS, the endothelial nitric oxide synthase. And the enzyme generates nitric oxide that on the one hand prevents platelet, monocyte, and neutrophil adherence, but importantly, diffuses also basolaterally to the muscle layers of the artery, controlling and relaxing muscle tension. So the question we asked ourselves, and many others have before us, is how do endothelial cells sense shear? How do they perceive the flow of blood along the surface of the endothelial cell? And to study this, we used a system that's analogous to the one others have used, and it's essentially a microfluidic chamber, which is shown here, where we can grow endothelial cells in culture, and we can subject them to shear flow by connecting the microfluidic chamber to a proton pump, or a liquid pump, where we can adjust the rate of flow. And normally we apply 15 dynes per centimeter square, which is similar to what's been estimated to occur in the arterioles of our circulatory system. So this is essentially replicates what had been done in the literature and shows you that we are able to do what other people have done as well. We grow endothelial cells under static conditions and they orient randomly. If we apply shear, now the cells become elongated and tend to align along the direction of the flow of fluid. We can then stain these cells for actin and use software to identify the fluorescence of phalloidin, which we use to detect the actin. And the software can identify the orientation of the fibers with respect to the duration of the flow. We have software that not only identifies the orientation, but can color code it with pseudocolor, and we can then quantify the randomness or orientation of the fibers with respect to the flow, where 0 degrees is the direction of the flow. And you can see that under static conditions, the main axis of orientation of the cells and their fibers is fairly random. If, however, we apply shear flow in one direction, most of the cells now align parallel to the direction of the flow. We can quantify the data, and you will see that there is a clear distinction between the cells under flow conditions and under static conditions. Now, what is it about the cells that senses the flow? And obviously, one would imagine that it's the portion of the cell that's in closest contact with the lumen of the vessel. Now, what I'm showing you here are two different magnifications of electron micrographs of blood vessels visualized after staining. And the particular staining that was used here highlights the presence of the glycocalyx. I won't get into the details, but suffice it to say that the cells look very hairy. And if you zoom in, they show this very pronounced glycocalyx. And in fact, the thickness of the glycocalyx is greater than the thickness of the endothelial cell. And it stands to reason that it would be the glycocalyx that would be most sensitive to the flow of blood in the lumen. Indeed, you can imagine the glycocalyx as the sail of a ship that is intended to sense the wind that's flowing along the vessel. So this is a schematic representation of what I mean here. This would be the endothelial cell, and this would be the glycocalyx. And clearly the flow would affect the glycocalyx much sooner and much more pronounced than it will affect the cell itself. So does the glycocalyx actually sense the flow? Is the glycocalyx required for the alignment of the cells and for the calcium changes that produce nitric oxide? Well, the main component of the endothelial glycocalyx is actually a glycan called hyaluronan or hyaluronic acid. And it's very easy to test the involvement of hyaluronan if we digest the hyaluronan using hyaluronidase. So what I'm showing you here are now cells in both cases treated under flow, exposed to either just a vehicle, in which case they align as we described before, very nicely, compared to cells that were pre-treated with hyaluronidase to remove the hyaluronic acid prior to the application of the shear flow. And you can see clearly that these cells are now oriented randomly, failing to sense the direction of the flow. And that is summarized in the graph to the right. Application of hyaluronidase prevents the cells from aligning after sensing the flow. I mentioned also that calcium goes up and signals the activation of eNOS. What I'm going to illustrate here is the flow-induced increase in cytosolic calcium. And the way we monitor calcium is by transfecting the cells with a calcium-sensitive fluorescent protein called GCAMP6. So prior to culturing the cells in the microfluidic chamber, we have transfected them with GCAMP6, which is shown here by its green fluorescence. Now, the image that I'm showing you here was taken prior to the application of the flow. When the movie starts, you will see a little white dot appear around here, which illustrates the moment when we apply a flow across the chamber of 15 dynes per centimeter square. So I'll turn the movie on. Oops, let me go back and try again. There's the movie running. It's looping, so you can see it time and again. So you can see when the dot appears, fluorescence goes much, much brighter, indicating that the calcium in the cytosol has gone up greatly. And we can quantify the fluorescence as a result of the application of shear. Now, what's important here is that we're comparing two different conditions. The control conditions, where the calcium increase is very obvious, with cells that have been pre-treated with hyaluronidase to remove the hyaluronic acid. And you can see that in the absence of hyaluronic acid, the application of shear flow fails to increase cytosolic calcium. So in summary, hyaluronic acid, which is the sail that senses the wind, is required both for the alignment of the cells and for the increase in calcium that results in the production of nitric oxide. So there's the glycocalyx. We know now it's important for sensing the flow, but this is the sail, and the sail is not going to move the boat unless the sail is attached to a mast. So how is the glycocalyx attached to the endothelial cell? So that when you blow the wind or when you apply the flow, you change the cytoskeletal organization and you induce the flow of calcium into the cells. And the mast that attaches the glycocalyx to the body of the cell is a hyaluronan receptor called CD44. It's a transmembrane protein that on the extracellular domain binds directly to hyaluronic acid and is attached firmly to the membrane of the endothelial cell. So what do we know about CD44? Well, we know that it is a transmembrane protein, single spanner, binds hyaluronic acid on the outside, and we have very good antibodies to detect the extracellular domain. When you do that, and you apply the antibodies to the cell and you stain the cells, you find a very peculiar distribution. What we're showing here is the distribution of CD44 by comparison with a membrane marker. Not important what the nature of the marker is, but this is a non-denominational generic marker of the membrane that illustrates every plasma membrane in the cell. This is CD44. And I think what should be obvious looking at these images is that the density of CD44 is much greater in the apical or luminal membrane than it is in the basolateral or bottom membrane. If you color code the images, if this is red and this is green, it'll be obvious that the top membrane is yellow, whereas the bottom membrane is virtually all green. So CD44, the receptor for hyaluronic acid, is largely localized to the apical membrane of the endothelial cells. So what keeps CD44 predominantly localized in one of the membranes of the cell? Well, there are two obvious possibilities. One is that it is immobilized on the apical membrane. The other one is that it is mobile, but it cannot traverse the junctions that separate the apical membrane from the basolateral membrane. So how can we tell if the molecule is mobile or immobile? Well, we did it by directly assessing the mobility of individual CD44 molecules. And for that, we generated from that antibody that I mentioned before, Fab fragments. You need Fab fragments in order to avoid cross-linking. So these are single epitope binding fragments of the antibody that will bind to the head group of CD44. We then covalently attached biotin to the Fab fragment and use the biotin to bind to the complex quantum dots coated with avidin. Avidin binds to biotin, and quantum dots, as you all may know, are nanoparticles that are very brightly fluorescent. In fact, you can see the fluorescence of individual quantum dots illustrating the presence or the location of individual molecules of CD44 on this apical surface of an endothelial cell. Now, there's a feature of quantum dots, which is that their fluorescence blinks, parpadean. At any point in time, their fluorescence is blinking in place. Disregard the blinking and concentrate, please, on the overall movement of these particles when the movie starts. Whoops. Uh-oh. For some reason, the movie doesn't want to work. Movie was working very well before. Let me see if it's a pity because this is an essential part of the talk and this was working before. I hope I'm still sharing and you can see it.
Perfecto.
Okay, great. So if you can see it, each one of the particles is blinking in position, but for the most part, they're immobile. Occasionally they move, and then you'll see that they move and they stop. And this is better illustrated if you track any single particle. It's immobile, it moves, jumps and becomes immobile again, stays in position for a long time, moves a little bit longer, and then gets arrested again and stays. So there's something that is trapping CD44 molecules most of the time in a single position. Now, let me go back and hopefully this will work. So we can use software to actually track individual molecules over time. And each one of these spaghetti strands shows a single molecule over time. And you can see that the molecules sometimes are immobile and stay in place virtually during the whole movie. Other times they move and then they stop. They move and then they stop again. You can see it by these dark blotches on the individual tracks. The software can actually identify different types of motion. We have molecules that are tethered or immobile all the time. They're shown here in red. There are other molecules or moments when the molecules are mobile, but they're confined. They move, but they move very little within a very constrained space. You can imagine this as a horse running within a corral. The horse is running, but it cannot go outside the corral. And you also have some occasional molecules that are freely mobile with Brownian motion. And we can quantify the different types of motion. So you have in the apical membrane, the membrane that faces the lumen of the vessel, about 40% of the molecules are immobile, about 40% of the molecules are restricted in their mobility, and only about 20% of the molecules are mobile. And interestingly, in the basolateral membrane, most of them are mobile, fewer are immobile. Okay, so if something is immobilizing CD44 on the apical membrane, one would naturally think that there is an anchorage site inside the cell and that the cytoskeleton would be involved. And indeed, if you look at the bottom of the membrane by scanning electron microscopy, this is one of the beautiful images published by Kuzumi and his group, you can see that the membrane is subtended by a very thick meshwork of actin filaments. You can actually see the actin monomers all along the fibers, and you can see that they're very dense and very highly connected, and it's easy to imagine that the cytoskeleton is responsible for immobilizing CD44. So that can be tested experimentally, and one way to do it is to extract the cytoskeleton selectively and to analyze whether CD44 remains attached to it. And for that purpose, we do extraction of the cells with a non-ionic gentle detergent in the presence of a cytoskeleton stabilizing buffer. This will dissolve most of the cell, but will retain the cytoskeletal architecture. If we then spin down the cells, we'll end up with a supernatant of soluble material and a pellet that contains the cytoskeleton plus cytoskeleton-associated proteins. And so if you do a Western blot and you look at actin, you will see that actin, some of it is soluble and ends up in the supernatant, but much of it goes to the pellet. CD44 is also predominantly in the pellet together with actin. So what this implies is that indeed there is an association between the cytoskeleton and CD44. That's validated here. CD44 that binds to hyaluronan crosses the membrane and binds to the cytoskeleton. Now, I hope you can see what's, oh, there we go. I hope you can see what happens when you now solubilize the actin using latrunculin A. Latrunculin A binds to actin monomers, prevents it from polymerizing. And when that happens, when you now solubilize the cells, virtually all of the actin goes into the supernatant, none remains in the pellet. Surprisingly to us initially, the pellet still contains CD44. The implication is that it cannot be actin that is retaining CD44 in the pellet. So, if it is not actin directly, what else could be holding CD44 onto the cytoskeleton and the pellet? And what we know is that the cytosolic domain of CD44 binds specifically ankyrin and ERM proteins, esrin, rhodixin, myosin proteins, which are all adapters between transmembrane proteins and cytoskeletal components. And in particular, we know that both ankyrin and ERM proteins can bind to spectrin. Spectrin is yet another type of cytoskeletal protein, which is illustrated here. Spectrin is made up of a heterodimer of alpha and beta spectrin that bind end-to-end, and they form long chains that are very flexible and attached by very short strands of very stable F-actin. And esrin, rhodixin, myosin, and ankyrin can all bind to spectrin. So it was entirely conceivable that through binding ankyrin and or ERM, CD44 would attach to spectrin. What does spectrin look like in the endothelial cells? And that is illustrated here, where we immunostain beta-2 spectrin in the same endothelial cells that I've been showing you all along, and yet again compare the distribution of esrin to a membrane marker. And very much like I showed you before, for CD44 itself, the apical membrane is greatly enriched in spectrin, whereas the basolateral membrane is depleted. So there's a good correlation between spectrin and CD44, and good anatomical reason for spectrin to be bound to CD44 via ankyrin or ERM proteins. So is it conceivable that spectrin binding is what dictates the immobilization of CD44 to the apical membrane? And that can be analyzed by silencing the expression of spectrin. siRNA can be used to silence the expression of beta-2 spectrin, and we know from existing literature that if you eliminate beta-2, then the alpha subunit of spectrin also disappears. So we can effectively silence the presence and expression of spectrin in the endothelial cells, and now proceed to analyze what happens to CD44. So we can go back to experiments like the ones I was showing you before. And again, this is a control with non-targeted siRNA. We're looking at quantum dots attached to the head group of CD44. And as before, it's pretty immobile. It's blinking, the occasional molecules jumping around and getting immobilized, jumps again, gets immobilized. Now, this is what happens in cells where you have eliminated spectrin with siRNA. And I think it should be pretty evident by comparing the left and the right panel that the mobility of CD44 increases greatly when spectrin is eliminated. And so at this stage, what we know is that the flow of blood pushes away the hyaluronan glycocalyx, that the hyaluronan glycocalyx is attached to CD44. And now we know that CD44 is kept immobile in the membrane by attaching to a submembranous meshwork of spectrin. So if we want to study the role of spectrin in sensing alignment, blood flow, and calcium, rather than doing siRNA on a regular basis, we decided to make CRISPR-Cas knockout clones eliminating beta-2 spectrin from endothelial cell cultures. And we have 2 separate clones that we analyze routinely, clone 4, clone 6. And this is validation that the silencing, or the knockout in this case, of spectrin is fairly complete. So now we can compare the behavior of wild-type spectrin-containing cells with that of knockout cells lacking spectrins. We grow either the knockout or the wild-type cells in our microfluidic chamber, as we did before, apply flux, and now we can study, as we did, alignment, calcium, nitric oxide. So, just as a reminder, static cells, whether they're wild-type or knockout, fail to align and align randomly or grow randomly. Wild-type cells, when you apply the flow in this direction, align in the direction of the flow very nicely. That can be quantified. If you now apply the same shear force for the same duration to cells lacking spectrin, they grow randomly and they fail to align. And that can be quantified, as shown here. And the only instance in which we see alignment of the cells along the plane of the flow is when spectrin is present, but not in the absence of spectrin. What about calcium? I showed you before this movie here, where I showed you that upon application of flow, which is going to be shown again by the white dot here, the calcium goes up very distinctly. There it is again. What happens now to spectrin knockout cells? So as before, the movie starts in the absence of flow when the white dot here appears here. The flow is being applied to the cells. Nothing. Now you apply the flow and nothing happens. So the ability of the cells to sense the flow depends not only on the presence of hyaluronan, but also on the presence of spectrin. As before, we can quantify the data. This is the normal increase in calcium sensed by GCAMs. This is what happens, This is what happens in the knockout cells devoid of spectrin. Application of shear fails to increase the calcium, and the data are fairly conclusive. You don't have to be a statistician to tell that this is significantly different from that. Okay, so we have another piece of the puzzle. Flow, the sail, the mast that attaches the sail to the boat, and the mast itself is attached to spectrin. But how does spectrin now relay the information to the calcium channels or to the cytoskeleton? Is spectrin itself sensing the shear force? And we can analyze how spectrin is reacting to the application of flow extracellularly by looking at the structure of spectrin itself. So spectrin, if you're not familiar with its structure, is a very long molecule consistent of a large number of spectrin domain repeats. And these repeats are actually alpha helices that go back and forth and are extremely flexible, such that if you apply tension on the molecule, this operates like a spring and stretches out. And this is what gives spectrin its unique flexible sensitivity. How can we tell if we are stretching spectrin? So what we did for that purpose is we used a construct that within one of the spectrin repeats has built in 2 fluorescent proteins, a blue fluorescing protein, cerulean, and a yellow fluorescing protein, venous. Now, when these 2 are closely opposed to one another, the fluorescence of cerulean activates the fluorescence of venous. You get fluorescence, resonance, energy transfer. So you excite blue, you get emission of the yellow. If you, however, stretch the molecule by applying tension, you separate cerulean from venous such that there's no longer a transfer of energy between the blue and the yellow. And what you get now is less fluorescence of yellow and more fluorescence of the blue. And in that fashion, what you've devised is a stretch sensor that can tell you about the state of stretching of spectrum. So these images here, again in pseudocolor, illustrate the fluorescence of cerulean and of Venus, either under static conditions or under shear. And you can see under static conditions, you excite cerulean and you get quite a bit of emission from Venus. Very good energy transfer. If you now apply shear to the cells, the fluorescence of cerulean actually goes up and that of Venus goes down, which means the resonance energy transfer between the blue and the yellow has gone down, implying that the distance between them has increased, meaning that you have stretched spectrum. You can quantify the data. This is the energy transfer under static conditions. This is under shear. Very importantly, if you pre-treat the cells with hyaluronidase, the sensor of the shear flow, you now fail to undergo the loss of energy transfer. So the stretching of spectrum induced by flow requires the presence of spectrum on the ventral side of the plasma membrane. So, yet again, I don't want to be tedious, but to emphasize it, this is the wind, this is the flow, this is the sail, this is the mast. Now, spectrum is attached to the mast, and spectrum stretches when the wind or the flow moves CD44 along the surface of the cells. So, does spectrum influence the shear at the plasma membrane itself? If you're going to alter the behavior of channels or the cytoskeleton, spectrum has to affect other components. And the way we measure that is yet again taking advantage of fluorescence. And we use a probe recently introduced by a Swiss group that's called FLIPPER. It's a very ingenious probe, also an energy transfer probe that has two components, two fluorophores that are built in a right angle to each other, connected by a flexible bond. When the angle of orientation between the two fluorophores changes, the efficiency of energy transfer between one and the other fluorophores is altered as well. And the angle between these two will be affected by the lateral tension of the membrane. If you squeeze the membrane laterally, now both fluorophores will align in parallel fashion, and the efficiency of energy transfer will change. And so we incubate the cells with FLIPPER. FLIPPER inserts into the membrane in between the lipid tails of the bilayer and will sense the tension between lipid moieties by altering its energy transfer. We measure the energy transfer by using fluorescence lifetime imaging. I can give you the details if you're interested, but suffice it to say for now that we use frequency domain fluorescence lifetime imaging to measure the energy transfer. And what we find is that you can tell when the stress of the membrane changes by looking at the intensity of the fluorescence emitted. And as a proof of principle, we change the intensity or the tension on the plane of the membrane by applying a hypotonic swelling to the cells. This induces stretching of the membrane, changes lateral tension, and as you can see clearly, changes the efficiency of energy transfer. That can be plotted. But most importantly for us, what we wanted to see is whether the tension in the plane of the membrane changes when we apply shear. And the answer is already shown here. Application of shear alters the fluorescence lifetime of the FLIPPER probe in a manner suggestive of a change in the tension in the plane of the membrane. Importantly, if you treat the cells with hyaluronidase, the change is minimized. Okay. And so at this stage, we know that as spectrin stretches in response to the changes in flow, spectrin will itself alter the tension in the plasma membrane, possibly affecting the permeability of the membrane to calcium. How would the membrane sense changes in tension? Well, it's no secret that most biological membrane, most cells, have mechanosensitive ion channels. And these ion channels respond to changes in the lateral tension of the membrane. And indeed, endothelial cells are one such cell. When you apply shear to the cells, we produce the calcium change that I've shown you before. If you apply GS-MTX4, an inhibitor of mechanosensitive channels, you completely prevent the changes in calcium. So clearly, endothelial cells possess mechanosensitive channels. And specifically, they were shown earlier to express piezo-1. Piezo-1 is the quintessential calcium permeable mechanosensitive channel. And we know it is present in the endothelial cells because piezo can be activated specifically by a molecule called Yoda-1. This has resemblance to Star Wars. When you apply Yoda-1, it binds to piezo-1, activates and opens piezo-1, and allows the entrance of calcium into the cell. Now, unlike this experiment where we open the channels by applying shear, here, this is static conditions. No flow is applied, but we introduce Yoda into the solution. And Yoda is sufficient to activate the inflow of calcium in a manner that resembles the effect of shear. But most importantly, not only does the effect of Yoda demonstrate that we're dealing with activation of piezo, but in the absence of spectrin, Yoda fails to activate the influx of calcium. So how does spectrin regulate piezo? It's conceivable that maybe in cells that don't have spectrin, that piezo-1 is removed from the membrane. But that seems not to be the case. If we take piezo-1 antibodies and we assess the expression of the channel in the cells in the presence and in the absence of spectrin, we notice that the level of expression of piezo-1 is maintained in the absence of spectrin. So what else could be wrong with piezo? Well, we know that piezo preferentially associates with curved microdomains of the membrane. And in fact, the reason piezo is so sensitive to changes in membrane tension is that it is a slightly curved molecule. It prefers to be in curved areas of the membrane. And when the membrane is stretched, you alter the conformation of piezo that then opens up and allows the entry of calcium. And one of those curved areas of the membrane that piezo particularly likes to localize in are caveolae. Caveolae are flask-shaped depressions of the membrane generated by the presence and assembly of caveolin 1 together with cavin. And we tested whether piezo-1, in fact, co-localizes with caveolin. Needless to say, the cell type that is most abundant in caveolin and caveolae are endothelial cells. So what is shown at the bottom are immunostains simultaneously of piezo-1 and caveolin on endothelial cells. What you can see is both of them show a distinct punctate distribution. And when you overlay them, the superimposition of the green and the purple gives you all of these white dots. There's very good colocalization between piezo and caveolin suggesting that the channel, for the most part, resides in the curved areas generated by caveolae. So what happens to caveolae when we don't have spectrin? And that is best analyzed by electron microscopy. So this is transmission electron microscopy of wild-type cells, looking at the apical membrane of the wild-type endothelial cell. And what you can see, and that's designated here by the asterisks, are individual caveolae that are well-defined depressions of the membrane, which are uncoated, unlike clathrin-coated bits. So multiple caveolae at short intervals on the surface of wild-type cells. You do the same thing in the knockout clones lacking spectrin. You'll see that the density of caveolae is greatly reduced when you don't have spectrin, as if the presence of spectrin is essential to either form the caveolae or maintain the caveolae on the apical surface of the cells. That can be, again, quantified over extensive areas of apical membrane from transmission electron micrographs, and you can see that the density of caveolae is much greater in wild-type cells than it is in the knockouts. So, in summary, what we think is happening is that you have a very interesting and complex interaction that manages to convey the signal from the luminal space of the vessel to calcium channels in the plasma membrane. And how it works is that you have a dense hyaluronic acid glycocalyx that is ideally suited to sense the flow of fluid along the vessel. This hyaluronic acid is attached to the cell, attached to the mast of the ship via CD44, the receptor for hyaluronan. CD44 itself is a single-spanning transmembrane protein that on its cytosolic domain attaches via ankyrin and ERM proteins to the cytoskeleton, but particularly to the to the spectrin cytoskeleton via ankyrin and ERM. Spectrin, being long, flexible molecules, are uniquely localized along the apical membrane of endothelial cells, and they're uniquely poised to sense movement because they have repeat domains that can stretch and can sense tension applied to the extracellular space via CD44. Stretching of spectrin will in turn stretch the membrane, apply tension on the membrane, such that as spectrin stretches, the membrane is also extended, enabling piezo1 to conduct calcium and signal the production of nitric oxide. And of course, I just talk about experiments. I don't do any. The person that did the bulk of the experiments is a brilliant former student that's now a doctor, Dr. Sivakami Milvaganam, in collaboration with my colleague Spencer Freeman, with some help from Ren Lee. And these are the agencies that supported the work, and I'll be happy to try to answer any question. Muchas gracias por su atención.
Muchas gracias, Sergio, por una plática muy bonita. No sé si...
¿Alguna pregunta? Ahí hay una.
Hola, Sergio. Muy bonita charla, la disfruté mucho. Soy Adán Guerrero.
¿Qué tal?
Va en español la pregunta. ¿Cuál es el tamaño de las invaginaciones que forman las caveolinas?
Aproximadamente 150 nanómetros.
Entonces cabe más que un solo canal iónico ahí. Pueden caber un grupo de un nanodominio funcional de canales iónicos.
Sí, no sé exactamente cuál es el tamaño de piezo, que es una molécula bastante grande, es una molécula enorme, pero efectivamente seguro caben más de una molécula.
¿Alguna otra pregunta en el auditorio?
Yo puse una en el chat.
Ah, sí, ahorita va a hacer una... Sí, un momento.
Tengo otra pregunta, Reta. ¿Este Flipper es comercial o lo obtuviste por colaboración?
No, Flipper es comercial ahora. Creo que lo vende la compañía Cytoskeleton, pero si no es Cytoskeleton, mándame un email y te digo dónde lo conseguimos, pero seguro es comercial ahora.
Muchas gracias, buenísimo.
No hay por qué.
La pregunta de Beto, ¿la quieres leer directamente? Sí, creo que es más
Como te
fácil.
digo, ¿piensan que es directa la interacción entre espectrina y piezo 1 o es indirecta?
No tenemos ninguna evidencia ni en favor ni en contra. Mi impresión es que probablemente sea indirecta. Se sabe que en el cuello de las caviolas hay interacción de proteínas del citoesqueleto que mantienen la forma de las caviolas. Entonces, la impresión que tengo es que en esa zona conecta la espectrina con el cuello de la caviola y cuando la espectrina sufre la extensión es cuando se activa piezo.
Muy bonita plática, gracias.
No hay por qué.
¿Alguna otra pregunta? Aquí hay una pregunta.
Sí, muchas gracias por la plática. Estas propiedades se pueden ampliar a todos los tipos de células endoteliales, en particular a las del cerebro.
Buena pregunta, no tengo una respuesta. Obviamente vamos a saberlo solamente cuando funcione. Lo que es seguro es que el shear flow, el stress que se aplica es mayor en las arterias y en las arteriolas que en los capilares y en la circulación venosa, porque la presión arterial va bajando. Entonces, el efecto del flujo va siendo menor. Eso refiere exclusivamente a la percepción del flujo. Pero eso no quiere decir que otras células endoteliales no tengan los canales y no tengan la manera de percibir mecanostress. Entonces, algún otro tipo de estreses mecánicos, extensión, etcétera, se podían percibir de la misma manera aún en otras partes de la circulación.
Gracias.
Por nada.
Sí.
Gracias, Sergio. Muy bonita plática. ¿Esto se podría extender a células musculares, por ejemplo, diferentes estreses que, según lo que entiendo, si estiras la membrana, activas estos canales de calcio que finalmente cambian la disposición de la célula?
Sí, absolutamente sí. Y no solo existe piezo como canal mecanosensible, hay toda una familia de canales de la familia TRP, Transient Receptor Potential, que también son mecanosensibles, tienen selectividades diferentes y sensaciones diferentes, y la expresión es diferente en los diferentes tejidos, etc. So, mecanosensation está presente probablemente en todas las células, en todos los tejidos, y probablemente incluso en membranas intracelulares. Algunos de estos canales están bien descritos en membranas intracelulares que continuamente están sufriendo rearreglos, tubulación, etc. Entonces, ese es un campo relativamente nuevo y muy importante, ¿no? Cuando un músculo se estira o cuando tenemos un impacto o algo así, siempre hay mecanosensación y son estos los canales que están involucrados.
Muchas gracias.
Bueno.
Sergio, como son expertos en lípidos, ¿saben si en esa región hay especialización y cambios en lípidos cuando ocurre el flujo?
Sí, muy buena pregunta también. las caviolas tienden a formarse en regiones que son ricas en colesterol. Entonces, hay especializaciones locales. Lo que nadie sabe todavía es si hay cambios agudos en la distribución de lípidos en el plano de la membrana o a través de la membrana cuando todo esto sucede. El colesterol, por ejemplo, se sabe que puede hacer flipping de una monocapa a la monocapa opuesta continuamente. ¿Qué pasa cuando aplicas tensión? ¿Qué pasa cuando jalas la membrana en cierta dirección? No sabemos.
Yo tengo una pregunta. ¿Hay alguna hipótesis que se trabaje del por qué se pierden los caviolis en la ausencia de la espectrina?
Una de las posibilidades que consideramos es que las caviolas aumentan su movilidad y se vayan a la membrana vasolateral, y eso en parte es cierto. Las caviolas se sabe también que se pueden desprender de la membrana y formar membranas internas. Entonces es posible que cuando pierden el anchorage, cuando pierden la conexión con el citoesqueleto, muchas de ellas se vuelven intracelulares. Y de nuevo, no lo hemos estudiado en detalle, pero se podría cuantificar el número de caviolas en la membrana vasolateral comparado con la membrana apical y las internas comparadas con las superficiales, pero eso es enteramente posible que se puede hacer.
Bueno, lo que entendí muy bien es filamento de actina.
Qué bueno que alguien entendió algo bien.
Ese es filamento de actina y shear stress es misma dirección. Entonces, y filamento de actina no es elástico. Entonces, células expande dirección perpendicular. Ese es un escenario, como es, imagino que venas como flexible, ¿no? Cuando pasa hemoglobina y tiene que expandir y regresar. Entonces, flexibilidad debe de ser perpendicular de dirección de actina.
Los filamentos de actina no son particularmente flexibles. La espectrina es flexible. Lo que puede confundir acá, y nos confundió a nosotros por principio también, es que el alineamiento de la actina en la dirección del flujo parece sugerir que es la actina la que está percibiendo la dirección del flujo, pero resulta que la actina que se está reorganizando es fundamentalmente la actina de los stress fibers en la membrana opuesta. Es una respuesta secundaria e indirecta, y esa actina es mucho menos flexible. Entonces, lo que parece que está sucediendo es que el esqueleto de la membrana apical, que tiene menos actina, pero tiene más espectrina, es de diferente tipo. Es un esqueleto más flexible, parecido al esqueleto de las neuronas. Las neuronas en el axón tienen anillos de actina comunicados por bandas de espectrina, lo cual le da flexibilidad al axón. Entonces, lo que sucede es que cuando cambia la señalización a través de la espectrina y del calcio, etcétera, etcétera, se rearregla la formación de stress fibers en la membrana opuesta de la célula. ¿Cómo sucede eso? No estamos seguros. Tenemos una evidencia preliminar de que vimentin, que es un componente de intermediate filament, puede estar pasando la señal de la membrana apical a la membrana vasolateral, pero hay un sistema complicado transcelular de señalización que permite la señal apical traducirse en realineamiento de los stress fibers en el lado opuesto de la célula.
Ok, más complicado de lo que yo pensé. Gracias.
The last question is on YouTube. Perdón. ¿Alguna idea acerca es del doctor López Casillas, alguna idea de la naturaleza molecular de la interacción con hialuronidasa o con hialuran?
Con hyaluronan, sí. El sitio de interacción entre hyaluronan y CD44 está bien caracterizado. Es un receptor y es específico. Hyaluronan es un polímero de dos azúcares, glucuronic acid y glucosamine, si recuerdo bien. Y la parte extracelular de CD44 tiene sitios de reconocimiento específicos de los azúcares de hyaluronan. Y es el mejor caracterizado de los receptores de hyaluronic acid.
Okay, we're going to move to the next speak.
Muchas gracias. Saludos.
Saludos, Beto. No hay por qué.
Gracias. Now, I'm very, very pleased to introduce Dr. Nicole King from the University of California. She's an evolutionary biologist recognized for her work on the emergence of multicellularity. Her research group developed the use of choanoflagellates, which are the closest living relatives of animals, as model organisms for genetics, genomics, and cell biological research. And her work uncovered genes essential for animal multicellularity and cell differentiation, and revealed some of the mechanisms that control the transition to multicellularity. Nicole King graduated from Indiana University with a degree in biology and from Harvard with a PhD in biochemistry. She was a postdoctoral scholar in evolution at the University of Wisconsin-Madison and joined the faculty of the University of California in Berkeley in 2003. She was awarded the MacArthur Fellowship in 2006, and later she was appointed as a Howard Hughes Medical Institute investigator. She was elected as a member of the Academy of Sciences in France in 2021, and recently this year she has been distinguished as a member of the U.S. National Academy of Science. Dr. Nicole King, thank you very, very much. It is a privilege to have you here.
Thank you. It's a real pleasure to be speaking in your symposium. I have to say that I've been an admirer from afar for your institute for a long time, and I'm just thrilled to be able to contribute to this symposium. So I'm really excited to talk about some of our most recent work. I don't know if it works to take questions throughout, but if any questions come up during my talk, I'm happy to take them. And of course, I'll be happy to take questions at the end. So in my laboratory, we are particularly interested in questions of animal origins. And I think we can all look around in our natural world and be astounded by the incredible diversity of multicellular forms that we see in the animal world. So we can see animals that swim, that crawl on the earth, and that fly, and they are eating plants or animals, other animals, or bacteria. They have many different ways of surviving in the world. And yet, all animals share some important things in common. One of the most important being that they all start their lives as a single cell. And so some of the mystery about animal biology really comes from trying to figure out how you go from a single cell to a complex multicellular organism in every generation. But in my laboratory, we're interested in a different problem of going from being single cell to multicellular. And that has to do with the origin of animal multicellularity. So we know that all animals are what we call monophyletic. They form a single group, and that single group had a single common ancestor whose biology we can try to reconstruct. And in my laboratory, we're particularly interested in understanding how that ancestor of animals first evolved. In particular, how did multicellularity evolve along the animal stem lineage, and how did mechanisms for regulating cell differentiation and the coordination of function occur? Now, ideally, we would want to try to reconstruct animal origins by looking to the fossil record. But the fossil record, as rich and wonderful as it is, does not actually tell us much about animal origins because the ancestors of animals were small, they were soft, and they lived somewhere on the order of 750 million years ago. And so even if we could find something that was a progenitor of animals, we probably wouldn't know it. And it wouldn't tell us anything about the underlying cell and molecular mechanisms that led to animal origins. So instead, we're focused on using living organisms to try to reconstruct the past. Just as historians look at documents that were written recently and try to reconstruct the past, we're doing the same thing with organisms. And in particular, we're focused on a group of organisms called the choanoflagellates, and for short, we call them choanos. And these are a group of microscopic eukaryotes that are the closest living relatives of animals. Choanoflagellates are as diverse genetically as animals, and we can compare their biology to each other to reconstruct the biology of their last common ancestor, and then, compare the biology of these reconstructed organisms as well as living organisms to try to understand the biology of their last common ancestor. Now, if you haven't heard of choanoflagellates before, they're really fascinating organisms. The cell body is about 5 microns, so about the size of a yeast cell. And at the top of each of these cells is a collar of actin-filled microvilli. And in the center of this collar is a long flagellum. And you can see this better in this living cell here, imaged using high-speed video microscopy. And you can see that the flagellum beats back and forth. And this is important because it creates water currents that pulls material out of the water column up against the collar. And the reason this is important is that actually choanoflagellates primarily feed on bacteria from the water column. And you can see one of these bacterial cells here for scale. So an essential part of choanoflagellate biology is swimming around, which what they do with the benefit of the flagellum, but also generating these water currents to pull bacteria into that collar. Now, when I started studying choanoflagellates as a postdoc, there were no choanoflagellate laboratories. And as I showed you, choanoflagellates are incredibly diverse. And so there was a real question of which choanoflagellates focus on in terms of developing a model, and how are the cells actually adhering into this form. Now, in pursuit of understanding molecular and cellular mechanism in choanoflagellates, we've spent a great deal of effort and time on trying to develop new tools. And I share this with you because we want more people to study choanoflagellates, and I want you to have a sense of what's now possible. So even though choanoflagellates were first described in 1866, very little laboratory research was done on choanoflagellates until just the last couple of decades. So in 2008, my lab published the first choanoflagellate genome. This is M. brevicalis. But then we really shifted our focus to S. rosetta, a different choanoflagellate. And since then, we've established methods for developing cell state transitions in this choanoflagellate. We've established forward genetics and the ability to do genetic screens. We've established mechanisms for transfecting this choanoflagellate and expressing tagged proteins. In collaboration with, Pavel Burkhardt, we've published a 3D ultrastructure of S. rosetta, and most recently have established methods for doing genome editing in S. rosetta. And this has been a really important leap forward in terms of the tractability of this organism. So one of the things that first motivated me to study choanoflagellates was to try to understand the genetic landscape or the genomic landscape within which animals first evolved. And what we've found is that, in fact, there are a large number of what we call animal innovations. These are genes that evolved on the animal stem lineage. And these include genes that many of us have studied in pursuit of understanding developmental biology of animals. And so these include Wnt, frizzled, beta-catenin, classical cadherins, etc. But I think what was really remarkable when we first started studying the genomes of choanoflagellates was that many of the genes that we best understand in the multicellular context of animal biology were actually present in the last common ancestor of choanoflagellates in animals, and therefore likely first evolved in a unicellular context. And these include things like C-type lectins, cadherins, and integrins. And I'm going to be particularly focusing on G-protein coupled receptors later in this talk. So what I'd like to do during my talk is first to introduce you to the life history of S. rosetta, and then tell you two really surprising tales, at least from my perspective, surprising, in that we've found that bacteria actually regulate key life history transitions in S. rosetta. This includes mating and multicellularity. And I'm going to close with some very new work from the lab. Really, these results are just coming in week by week. It's been very exciting, in which we've discovered, I think, some really interesting and unexpected things about how GPCRs are functioning in a choanoflagellate. Okay, so first let me start by introducing you to the process of colony development in S. rosetta. And you'll see in a moment that actually S. rosetta can live quite happily as a single-celled organism that divides and produces other single-celled progeny. But in response to cues that you'll learn more about in a moment, it will switch into a rosette development pathway. I started that movie too soon. And in this rosette development pathway, it will divide along the apical basal axis, and now the two daughter cells will remain attached, and they'll continue dividing with the cells remaining attached until they develop into this three-dimensional rosette form. And so here is a video in which you'll see a single founding cell dividing over and over again to produce a rosette. So the key take home here is that all of these cells are related. They all have the same genome. And this is if you think about it, a transition to multicellularity, but it happens in the life history of the choanoflagellate. It's not this evolutionary transition that is our main focus. Now, the fact that these cells are related is actually borne out when you look at the ultrastructure of their interactions. So neighboring cells in rosettes are joined by an intercellular bridge that's the product of incomplete cytokinesis. And you can see this here in this TEM section, in which you see that these two cells are connected by this cytoplasmic bridge, which has two thick electron-dense plates in it. And here again, in a zoomed out picture, you can see the cytoplasmic bridge. But in addition, I want to draw your eye to the fact that there's also extracellular matrix that's filling the inside of the rosette. So we wanted to understand what regulates this developmental switch. What determines if S. rosetta continues dividing as a single cell, or if it switches into a pathway that leads to the development of this multicellular form? And we are interested both in extrinsic factors as well as the intrinsic or genetic regulation of this process. But initially, we had little to no progress. And this is because although S. rosetta was isolated from nature as a multicellular rosette, when I brought it back to the laboratory, it actually almost never formed rosettes. And more typically, what we had were cultures of single cells like this. And hopefully in the background, you can see this granular material. This is the prey bacteria with which we have to grow S. rosetta. So this is sort of what the cultures looked like when I was studying them as a postdoc. And because it was recalcitrant to the types of approaches we wanted to take, that's why I initially focused on sequencing the genome of a different choanoflagellate, Monocyta brevicollis. So when I started my lab at UC Berkeley, we started treating this original culture with different combinations of antibiotics to try to reduce the amount of bacterial diversity. And we couldn't get rid of all the bacteria. We didn't want to because they needed to eat bacteria to survive. But we just wanted to change the bacterial diversity. And what was really surprising was that with a certain combination of antibiotics, we got cultures that looked like this. They had enough bacteria to survive, but they never formed rosettes. And so that raised some interesting questions about what regulates rosette development and if there might be an environmental component. Now, of course, it was possible that the antibiotics were the explanation, but even when we removed the antibiotics, we still never saw rosettes. And it could have been that they were starving, but in fact, their growth curve was great. It looked terrific. And so a third possibility was that there might be signals from environmental bacteria that in some way stimulated rosette development. And so to test that hypothesis, an undergrad in the lab, Rick Zuzo, started isolating individual bacterial strains from the original environmental isolates, and he started adding them into these cultures that never formed rosettes. And lo and behold, he discovered one particular bacterial species that alone was sufficient to rescue colony development. So this was a very exciting finding for us. So this bacterium was in a genus of bacteria that were relatively poorly studied, the Algorophagus bacteria, which I had actually at the time never heard of before. And this finding was really exciting because these bacteria were natural prey of S. rosetta. They were found in the environment in which S. rosetta was isolated, and they were related to bacteria in the vertebrate gut, members of the Bacteroidetes. And so the hope was that if we could understand the molecular mechanisms by which Algorophagus induced rosette development, we might learn something more generally about these types of host microbe interactions. And so to this end, we collaborated with a chemical ecology lab, John Clardy's lab at Harvard Medical School. And I won't go into all the details, but just say that we were able to isolate the compound made by the bacteria that is sufficient to induce rosette development. So we've named this molecule RIF1, rosette inducing factor 1. And this is a very interesting molecule because in fact, it is related to sphingolipids, which are important for signaling in animals. And what distinguishes this molecule from sphingolipids is that it actually has a sulfonic acid head group here. And that's important. If we replace that sulfonic acid head group, it doesn't work anymore. So, the finding that bacteria regulate this transition was exciting. The identification of a particular molecule was also very exciting. But what we additionally wanted to know was how are the choanoflagellates sensing the bacterial signals? Are there receptors and are they homologous with animal proteins? And I should say that a number of experiments led us to believe that there should be a receptor that we would be able to find. The most important being that, in fact, we've also isolated a small molecule inhibitor of rosette development. And that inhibitor looks like a hydrolyzed form of RIF1, and it behaves like a competitive inhibitor. So it looks like they're both competing to bind to a shared receptor ligand binding site. Okay, so there are a lot of approaches that you might want to take to find the receptor. Some of these are to tag this molecule, but it turned out that was not possible. Any tag on this molecule actually blocked its activity. And so instead, a very brave graduate student in the lab, Tara Levin, proposed to develop genetic screens in choanoflagellates. And at the time, I was actually, very excited about the idea. Having an organism that has forward genetics really opens up a lot of opportunities for doing unbiased screens for genes that are important for your phenotype of interest. But there were a lot of reasons to believe that it was going to be very difficult to get this working in choanoflagellates. And I was worried, about Tara working hard on this and it never ending up with any success. But I shouldn't have worried. She identified all the critical barriers to this working and she broke them all down. It was amazing. So the way this screen worked was that cells grown in liquid media were exposed to a chemical mutagen, either EMS or X-rays. And these mutagenized cells were then separated into individual clones by limiting dilution. And so in a 96 well plate, each well would either be empty or contain one cell. And these cells then would be allowed to propagate and would be induced to form rosettes by the addition of that bacterial signal that I told you about. And then Tara screened laboriously by microscopy to see whether the wells contained rosette-forming cells or cells that had some sort of defect in rosette development. And just to give you a sense of how hard this was, she screened over 36,000 clones, and from that only got 18 rosette mutants. But these were, really informative, and they fell into a number of different particular classes. So here on the far left, you're seeing wild type cells. When they are induced to form rosettes, you get these beautiful rosettes. And when they're not induced, they can either be single-celled or form these multicellular chains. And so she found that there were a number of different phenotypic classes that she could recover through this screen. One particularly interesting one was class A. These can form perfectly normal chains in the absence of induction, but when treated with the inducing molecule, never formed rosettes. They remained in this chain-like form. The others tended to have mutant defects both in the uninduced state and the induced state. And one of the most abundant phenotypic classes formed these big aggregative clumps, both in the presence of, or in the absence of mutagenesis, in absence of induction, or in the presence of induction. Okay, I just noticed that I have a question saying that the rosette-inducing molecule looks like a prostaglandin. Are they related? And I'm afraid I don't know what prostaglandin looks like, so I can't answer that question. But it's not something that's come up before. Okay, so going back to this rosette screen, we really wanted to know what were the genetic lesions underlying these mutations, but we had a big problem. So, and that problem was, how are we going to map specific mutations to phenotypes of interest? And the key reason why this was a problem was, first of all, we didn't have genome editing working at that point, but we also didn't know anything about the sexual cycle of choanoflagellates. No one had ever observed choanoflagellate mating. We didn't even know what it would look like. We didn't know if it could be regulated in the lab. If they did mate, we didn't know if we could isolate meiotic products. And we weren't sure how we would map genetic lesions. And Tara actually overcame all of these challenges. So even though I mentioned choanoflagellates have been studied starting in the mid-1800s, there had been no reports of choanoflagellates mating. So it was a little unclear what to look for, but Tara started reading the literature about mating in diverse microbial eukaryotes, and she found that a common thread is that microbial eukaryotes often start to mate in response to environmentally stressful conditions. And so she tested a number of different conditions and found that under starvation conditions, she could actually induce mating. So even though this induces mating, actually you see that mating occurs quite rarely. So in this particular field of view, in fact, there are only two cells that are about to mate, and that's the small cell here and the large cell. And under starvation conditions, mating typically occurs between two cells of different sizes. And so we've provisionally called these small cells the male gametes, and these large cells the female gametes. So if we zoom in, what you'll see is that these cells will reorient. And then this larger cell is facing this way with the collar extending in this direction. What you'll see is that the male cell attaches next to the collar, and then they fuse. So I'll show that to you. And that essentially is choanoflagellate mating. And so what we're finding is that the hallmarks of mating in these choanoflagellates are that haploid cells first need to attach The cells then fuse their membranes. The nuclei fuse, we get a diploid cell, and then through meiosis return to this haploid state. And so this finding that mating occurs in choanoflagellates was really cool because it expanded our understanding of their life history. So we already knew that a single cell actually sat at the hub of a life history that was rather dynamic. This single cell could divide and stay together to produce a rosette, or under other conditions could divide and stay together to form these chains. These cells could actually land and attach to substrate to form these thecate cells, and these could convert into small cells or return to this cell type. But now what we discovered was that there was another part of the life history that was involved in sexual reproduction, in which these small cells could fuse to other larger cells and then mix their genetic information and return into the asexual life history. So because of this discovery, I mean, not only did we find that, that the life history of S. rosetta was much more complex than we previously realized, but it also became possible to successfully map these rosetteless mutants. And so Tara focused on these class A mutants, and she was able to map one of them to a very special C-type lectin. This is a class of genes that binds to specific sugars. And in this case, the C-type lectin was part of the extracellular matrix. You can see it here in teal. And it actually sits at the base of the cells that are in the rosette, holding them together and giving important structural integrity to the rosette shape. And then Laura Wetzel focused on this class of mutants, and what she found, she mapped two of them, and they both mapped to a class of enzymes called glycosyl transferases. These also are essential for sculpting the extracellular matrix. And one of these you can see here is actually localized to the apical pole of these cells. And when these glycosyl transferases are disrupted, then you get this promiscuous clumping of cells, both in the absence of induction and in the presence of induction. And what I'm not showing you here is that if you shake the flask, all those cells fall apart. And what you find is that there's no evidence for a robust rosette development in this class of mutants. So these results were very exciting to us. It was the first evidence that we could map, we could isolate mutants by their phenotype and map them to genes. But it turned out that none of the genes that we identified so far through the genetic screen had the properties that suggested that they would be the receptor for the rosette-inducing factor. And so another graduate student, Ariel Woznica, decided to use a different approach. And in this approach, she proposed to use RNA-seq to try to find genes that are specifically upregulated, choanoflagellate genes that are upregulated when they are in the rosette form and not in the non-rosette form. But a challenge there is that when we expose S. rosetta to the inducing bacterium, in this case, Algoriphagus machipanganensis, the choanoflagellate is being exposed to a lot of bacterial cues, not just the rosette-inducing factors. And so there might be many different transcriptional changes, not all of them specifically associated with the detection of the rosette-inducing factor. But we had also discovered a number of other bacteria that could induce rosette development. And these are shown with X's here. And you can see they come from many different clades of bacterial diversity. So the ones with X's had been tested and were known to induce rosettes. And the ones without X's, even though they were in many cases closely related to rosette-inducing bacteria, did not induce rosettes. And so the idea was that this would give us a negative control. So that was the plan. But Ariel did something that other people in the lab hadn't done. And that is that when she tested the non-inducers, she looked after 24 hours, which is when you typically see rosettes, but she also looked right away. And when she looked right away, which was far too early for rosettes to form, she saw something really unexpected in cultures that were treated with Vibrio fischeri. So let me show you what she saw. On the left are choanoflagellates that have been treated with a control bacterium that causes no difference. And you can see these cells are just swimming around normally. But when treated with Vibrio fischeri, she found that these S. rosetta cells started to swarm. She would see these swarms forming, and then the cells would start to stick together. Okay, so this was very unexpected. And we weren't sure what to think about it, or was it even a physiologically relevant state for the choanoflagellate? But then Ariel started to realize that actually swarming is a mating behavior in animals and also in diverse microbial eukaryotes. So this behavior that leads to a high local concentration of individuals often precedes waves of mating and genetic exchange. And so she hypothesized that that might be what the Vibrio fischeri was unleashing. And in fact, that was correct. So when she treats S. rosetta with Vibrio fischeri, what she finds is that in contrast to cells that have been induced to mate by starvation, these cells actually are the same size. They pair up by the basal poles of the cells. The membranes fuse. The nuclei migrate and fuse together. And not only that, but she found evidence for recombination. And so this, I thought, was a very sweet experiment in which she took two strains that could be distinguished from each other by a small number of genetic variants, which are shown here. And when she treated with control bacteria, she found that she only recovered progeny that had one or the other of the parental genotypes. But when she treated with Vibrio fischeri, she found that the progeny actually had clear evidence of recombination, genetic recombination among the parental genotypes. And so this revealed that the Vibrio fischeri activity not only triggered swarming and cell fusion, but it also triggered recombination. And so this was our second very exciting piece of evidence that bacteria are regulating key processes in S. rosetta biology. And so this just summarizes what I told you. If you mix haploid cells with Vibrio fischeri that is producing this mating-inducing factor that we've called Eros, we find that this rapidly promotes swarming, promotes cell fusion, and then leads to diploidization, meiosis, and recombination. So, I've just told you two examples in which we were hunting for the receptor of the rosette-inducing factor, and we failed, but discovered something exciting in the meantime. The first being the genetic screen, which led us to discover genes that were essential for extracellular matrix formation, that were important for the three-dimensional integrity of the rosette. And the second example I told you was this RNA-seq approach. It started out as an RNA-seq approach and instead led to the discovery that Vibrio fischeri produces a protein in this case that induces mating. So this is going to be a theme. I particularly enjoy telling these stories to graduate students because sometimes you set out to study one thing and you discover something entirely different. And so those tangents, I think, were really important for the research in my lab. Now, I want to close by telling you one more story about how the hunt for the receptor of rosette-inducing factor 1 led us in an unexpected direction. And in the end, we still don't know what the receptor is, but I hope you'll find this last story interesting. This is something that's really fresh in the lab, and I'm very excited about. So, returning to this question that has been captivating for us, we decided one more time to try to find the receptor for the rosette-inducing factors. And the project I'm going to tell you about really arose because of the pandemic. And so this project was led by a new postdoc in the lab, Alan Garcia de los Bayanos. He started in the lab literally a month before everything shut down in the pandemic. And his plan was to study a new choanoflagellate that we had isolated that has very interesting ways of interacting with bacteria, including growing a microbiome inside the center of a rosette. And so that's what he was going to do. But then everything shut down. And everyone in my lab, like many of you, I'm sure, became a computational biologist. So Alan decided to focus on looking at the evolution of G protein coupled receptors. This is the largest family of cell receptors in animals. These G protein coupled receptors are typified by having 7 transmembrane passes, coupled with a few other diagnostic domains that I'll tell you about in a moment. But what's fascinating about them in animals is that they are capable of responding to a diverse number of extracellular cues, most importantly in this context, lipids. And in fact, the best known receptor for sphingolipids are G protein coupled receptors. And so remember, I told you that the rosette inducing factor 1 looks like a sphingolipid. And so we were curious about whether the receptor for RIF1 might be a G protein coupled receptor. So Alan decided to focus on this class of proteins while stuck at home during the pandemic. And in the process of learning more about GPCR diversity, became very focused on a specific class of GPCRs called the adhesion GPCRs. This is like a particularly good target because adhesion GPCRs actually mediate multicellularity and cell-cell adhesion in some contexts in animals. So these adhesion GPCRs have a set of conserved adhesion motifs in the extracellular portion. There's this cleavage site called GPS that's conserved, and then the typical 7 transmembrane domains. And when he looked at the GPCRs that were conserved across choanoflagellate diversity, which I'm showing you here, he found that the adhesion GPCRs represented the vast majority of GPCRs that were found in each genome. And here's our hero here, Salpinguica rosetta or S. rosetta, and you can see that it has 19 total GPCRs, and 12 of these are adhesion GPCRs. Now, if we step back and look across eukaryotic diversity, what we can see is that animals have a tremendous number of adhesion GPCRs encoded in their genomes, but so do many of their unicellular relatives, shown here. And that abundance of adhesion GPCRs really drops off as we get further away from animals. And that also made this an interesting group of proteins to focus on. Now, when he peered at the specific types of GPCRs that were present in choanoflagellates, there was one class that was particularly interesting because of how highly it was conserved in this group of choanoflagellates here that includes S. rosetta. Pardon me. So I'm going to tell you the story of his, once he was able to get back in the lab, his deep dive on studying one particular GPCR that we've now named Clumpy for reasons that I hope will be obvious soon. So this is the protein domain architecture of full-length Clumpy. And you can see that it has these EGF domains here and here. It has a LAMG domain. This is the cleavage site that I told you about before, and then the 7 transmembrane domains. And because we now have genome editing possible in S. rosetta, he was able to truncate the gene at places that encode for the end of the LAMG domain, and then even at the very amino terminus of the protein, so that all of these protein domains were deleted. And what I'll tell you right now is that all of the phenotypes he observed were the same in these 2 truncation mutants. So we think that these are actually knockouts. Okay. So the first and most striking thing that he saw was that in these two mutants, clumpy 1 through 553 and 1 through 95, they led to cell aggregation. So here on the left, you're going to see the behavior of cells with wild type clumpy, and then these, the middle and the right are the mutants. So hopefully you can see that over time, these two sets of cells clump up, but the wild type cells don't. And if you look across time and quantify, so here again is the wild type or full length. This is one of the mutants, and this is the second mutant. And you can see that over time, these two mutants form these large clumps, which you can see mapped along the y-axis. And so even at 30 minutes, it's most obvious, in which you can see you have lots of large clumps here, and you don't see any large clumps in the wild type. So that was the first and most obvious aspect of its phenotype. And that was really surprising to us because it's an adhesion GPCR, we thought that we would lose cell-cell adhesion. Instead, it seemed to unleash cell adhesion under conditions where we usually don't see cell adhesion. Now, the next question was, did it have results in any growth defects? And in this case, there were no growth defects at all. The cells grew normally, completely comparable to the control. Well, what about rosette development? Are there any defects there? So here on the top is wild type, so no induction. And when you do induce rosettes in wild type, you get beautiful rosettes. And now here, one of our mutants, if it's not induced, and here what he's done is to shake the flask so that those clumps break apart. The cells are all unicellular as expected. And when rosettes are induced, you get these beautiful rosettes. And again, this is completely comparable. So clumpy, something is unexpected here because clumpy is an adhesion GPCR, but you knock it out, you don't lose clonal multicellularity, and you get this aggregation behavior in the single cells. So the next question that Alon asked was, how exactly are these cells interacting with each other? And you'll remember that when mating was induced in the Vibrio fischeri example, the cells were interacting by the basal poles of the cells. But here with clumpy mutants, they're actually interacting by their collars. And so they form these aggregates, but instead of the cell bodies touching, it's the collars that are touching. And Alon quantified this by going through and essentially measuring the nature of these different interactions. And so the cell body interaction that you see in rosettes or in Vibrio-induced mating, these are actually quite rare. What's much more common are interactions between cell bodies and collars or between collars and collars. And you can see that mapped here. Well, that was, I think, starting to point Alon in the right direction because although the interactions between cells were not the same between clumpy and Vibrio-induced mating, in fact, it was very similar to the ways in which cells clump up when they're starved and when mating is induced by starvation. And so you can see that here. And when he measured the nature of the interactions in starved cells, as you can see here, he found that they were comparable in terms of the percentage of cell body collar interactions and collar interactions. So at that point, he really felt like a very reasonable hypothesis was that for some reason, clumpy might in fact be leading to essentially promiscuous mating under conditions when mating was not typically observed. And so to test that, he asked whether cell fusion was happening more frequently in mutant cultures than in wild type cultures. And to do that, he took strains that had been labeled either with MTFP in the cytoplasm or with M-cherry fused to a histone, H2B, in the nucleus. He mixed these wild type cells together for 20 hours and then counted the number of cells in which there were both fluorophores. And so you can see this cell that has staining in the cytoplasm and the nucleus, and you can see the overlay there. And so he established these tags in the mutant strain, and then asked if he did the same experiment over 20 hours, how frequently would he see cell fusion? And what I hope you can see here is the percentage of fused cells on the y-axis in the control, which is almost none, compared with clumpy. And we see nearly a 5-fold increase in the number of fused cells under non-inducing conditions in the clumpy mutant versus the control. So this suggested that the phenotype, the aggregative phenotype that we see in the clumpy mutant really is a mating phenotype, and that clumpy might be a negative regulator of mating. So the next thing that Alon did was to try to get some insight into the molecular pathways underlying the clumpy phenotype. And there's much more to this story than I have time to tell you about, but I just want to highlight a few examples. So when Alon looked at the transcriptional profile of clumpy cells versus wild type cells, he found that there were dramatic changes in gene expression, and that there was a small class of genes that was downregulated and a second class that was significantly upregulated. And interestingly, the downregulated genes were by and large retrotransposons, and their regulation is often tightly linked to early stages of gametogenesis. There were a larger diversity of genes that were involved in, or that were upregulated in response to the clumpy knockout. And in work that I don't have time to tell you, Alon has discovered that cyclic GMP signaling is key to this phenotype. In fact, he can reverse the aggregative behavior and cell fusion simply by adding exogenous cyclic GMP. But I want to focus on the cell adhesion and cell fusion category and let you know that one of the classes of genes that was specifically upregulated in response to knockout of clumpy is HAP2. This is a protein that is key to gamete fusion in diverse eukaryotes, shown here in red along the phylogeny. And it's involved in regulating gametogenesis, not only in diverse animals, but also in the microscopic eukaryote, Chlamydomonas reinhardtii, and in the ciliate Tetrahymena thermophila. And so this protein has an ancient and highly conserved role in mediating gamete fusion in diverse eukaryotes. It's conserved in choanoflagellates, and it's specifically upregulated in the clumpy mutant. And so those results taken together have led us to think that that clumpy, this adhesion GPCR, is actually acting as a negative regulator of mating that's induced by loss of nutrients. And our reason for thinking this is that if we look at wild type cells under conditions of high nutrients, we find that the level of collar adhesion is low, the level of cell fusion is very low, and the expression of HAP2 and other gamete genes is very low. In contrast, when we look at low nutrient conditions with wild type clumpy, we see changes on all of these axes. So we get a high level of clumping and collar adhesion, we get increased cell fusion, and we get increased gamete gene expression. And these features are actually unmasked when we simply truncate the clumpy gene. And so even under high nutrient conditions, if clumpy is mutated, we get all of these behaviors that are associated with mating and gametogenesis. And so I've told you a number of different stories today that have to do with how the dynamic life history of Estruzetta is really subject to a combination of exogenous cues like bacterial lipids or the mating factor from Vibrio fischeri, and in addition, nutrient levels. But we're also starting to gain some insight into the endogenous genetics of the choanoflagellates that help us understand how those cues get translated into the signaling pathways of the choanoflagellate. And so with that, I'm going to stop. I'd be very happy to take questions. And I want to thank my wonderful laboratory who really held it together during the pandemic, and we're excited to be coming out on the other side. Thank you very much. And then I should ask, how should I handle questions?
No problem.
Hi, very nice talk. Thank you.
Thank you.
That. Have you done any experiment by exposing the choano to B. fischeri and to see if there's a downregulation of clumpy, or is there any change in the expression of any other gene? Because maybe that increases the diffusion of the cells. No,
that's right. That's a great question. We haven't done that yet. In fact, all these results are extremely new. So I've loved talking about this and getting ideas from people. And that's one of the things that we're planning to do soon, looking at the expression of clumpy when in response to Vibrio fischeri.
Yeah, well, thank you.
Thank you.
There is a question. Can you read a question in your chat, or do you want me to?
Yes,
read?
I can see it. Are the characterized mutants able to go into the sexual cycle, and do they present two sex type cells? So I'm not sure. Does that mean the mutants of clumpy, or does that mean the mutants of I'm wondering if the question relates to these mutants. Let me.
Can I say?
Wait. There. Yeah.
Yeah, I refer to the first set of mutants that you guys isolated.
These mutants.
These ones.
Yeah. So these class D mutants.
Yeah.
So in work that I am not showing you, these actually adhere through their cell bodies, and they do not seem to go through fusion. So it does not seem like these are gamete cells.
Okay.
If that answers your question.
Yeah, and the cells, the small and the large cells are found in the different mutants. You have both types, sex types.
In these mutants, no. You might see this last class. We call these. This is solo mutants, and we know nothing about this. And a new graduate student is going to be exploring what these are. But these never become multicellular. I suspect they are not gametes because they never interact with each other. They're antisocial.
Yeah. Okay. Thank you. Great talk. Thanks.
Thank you.
One question. These male and female cells, are they sequenced? Do they have any genomic property that distinguishes or determines their sex?
So the problem we have, if you look here, there's very few cells that are actually mating. So even though there's a lot of diversity in cell morphology, it's difficult to identify which are the ones that are about to mate. And so we're still trying to figure out how to do that. In fact, having this clumpy mutant may help us kind of increase the amount of mating that's going on. So yeah, in terms of genetic loci, in work that I didn't tell you about, it looks like that we're actually upregulating genes that are associated with mating type, like a particular class of homeobox gene that acts as a mating type regulator in yeast, and also an HMG box protein. So we may have cryptic mating types that we haven't known to look for before. And so I think that's another future direction that we're going to be going after.
Okay, thanks. Another question is about is there a kind of evolutionary advantage in for like rosettes have some advantage?
Yes. So that's certainly been a matter of speculation in some work done by Mimi Cole, who's also at Berkeley. She found that the rosettes were able to escape predation by ciliates, whereas single cells are eaten by ciliates. Oh wait, I'm getting this wrong. Yeah, no, that's right. And then, but in contrast, it turns out that an amoeba can capture rosettes, but it can't capture single cells. So it seems like there are potential benefits under some conditions, but there are also trade-offs. So you can imagine why the choanoflagellate might have this dynamic life history, because it depends on who else is out there and what the environment looks like, whether it's better to be in one cell state versus another.
Thank you. There is another question in the, a couple of questions in the chat. Do you want me to read them, or you prefer to?
Sure, I can do it. I can do it. Okay, so one question we have here is, the cytoplasmic bridges from the incomplete cytokinesis resemble cytoplasmic bridges found in gamete formation in metazoans. For example, the cysts in the formation of Drosophila oocytes or meiosis during spermatogenesis. Do I think that a formation of a rosette is something like a primitive definition of germ cells? So I'll just return to that picture. So yes, they do share some features in common with ring canals, for instance, or other structures that form during gametogenesis. But there are some things that are different also. We never see microtubules extending between them. We also don't typically see mating in the context of rosette development. So I guess at this point, we just don't know. It certainly is, it certainly could be. It's an interesting thing to think about, but it's not a requirement for mating. And I think we're agnostic at this point about whether this might be something like germ cells. I can also answer, I hope that answered that question. I could answer the next question also. In terms of starving, changing many cell parameters, ATP, of course, how is metabolism related to clumpy? That's an excellent question. Frankly, we don't know yet. That's definitely something we're looking forward to exploring in the future. The fact that there are these two different pathways to mating, starvation versus Vibrio fischeri, and the cell biology is completely different in both contexts, is something that is rather mysterious to us, and we definitely want to try to understand better.
I'm wondering how choanoflagellate die. Do they have apoptosis or programmed cell death?
They definitely have the genes for apoptosis. There's a paper that was not from my lab, but explored the evolution of genes for programmed cell death, and choanoflagellates express them. That's not a phenomenon we've studied extensively, but we have, under certain conditions, seen cells exploding in culture. That's about the extent of what we know on that. I hope you can see that there's a lot that we have to learn yet. It's wide open.
Is there any evidence of recombination in the starvation-induced meiosis?
There is. I just didn't show it. But in fact, we were able to use recombination through the starvation-induced meiosis to build a genetic map for the choanoflagellate.
One last curiosity: how is are there chromosomes? Is there chromatin? How are histones? How is that related to higher eukaryotes?
Yeah, it looks. I mean, I think in many ways these are essentially free-living animal cells. They're very much like animal cells. They are they have chromatin, they have all the normal histone components that you might expect. They have telomeres. In those ways, I think they're very similar to a canonical chromatin context of an animal cell.
One other question you have in the chat?
Yeah, does the osmotic condition of the growth medium affect rosette formation? This isn't something we've explored a lot, but we have played around with it a little bit. And as far as we can tell, it does not affect rosette formation. That actually is a great opportunity, though, for me to say that when we've played around with it, it's been at the physiology course at the Marine Biological Laboratories. And I do want to just send out an invitation to any interested graduate students or postdoctoral fellows to please apply to that course. We love having students from all over the world. And it's a great opportunity to learn about a lot of really exciting new areas of research. So if that interests you, just look up the MBL physiology course and think about applying.
Okay, thank you very much. It's been this very beautiful talk, and thanks for participating in our symposium. Thank you.
Thank you. It's been a real pleasure. Enjoy the rest of your symposium.
Okay, bueno, por razones ahí de agenda de todos, quedó un poquito aislada la última plática, que es después de la comida, pero pues los esperamos aquí a las 4 de la tarde para presenciar la última plática del doctor Oliver. Nos vemos más tarde.
Gracias.